Here is something that will change how you see yourself tonight. Inside every single cell in your body, there is a gene that is broken. Not damaged, not weakened, broken, completely non-functional. A stretch of DNA that was once responsible for one of the most essential biochemical processes in animal biology, and it no longer works. It has not worked for roughly 40 million years, and it will never work again. But here is the thing. This broken gene exists in every human who has ever lived. Every person breathing right now. Every person who ever breathed before. Every child who will ever be born. The same breakage, the same shattered sequence. Passed down from parent to child for 40 million years, never repaired, never discarded, just carried. Like a cracked heirloom nobody throws away. The gene is called gulo, and in most mammals, gulo does something that seems almost magical when you first understand it. It synthesizes vitamin C directly inside the body, from scratch. Your dog makes vitamin C, your cat makes vitamin C, cows, horses, mice, elephants, whales. They all produce it internally on demand as much as they need, without ever eating a piece of fruit in their lives. It happens in the liver, in a 4-step biochemical process so ancient and so conserved that it has been essentially unchanged for hundreds of millions of years. In humans, that process stops at step 3. The 4th enzyme, the one encoded by GULO, does not exist. The gene is there. The structure of it is there, like a blueprint still rolled up on a shelf. But the instructions inside have been scrambled. The sequence is corrupted. The gene is what scientists call a pseudogene, a ghost, a record of something that used to work. And what happens when you don't get enough vitamin C from your food? Your body cannot synthesize collagen properly. Collagen is the structural protein that holds connective tissue together. Your blood vessel walls start to weaken. Tiny hemorrhages appear under your skin. Your gums begin to bleed. Old wounds that had healed years ago reopen. Your joints swell. Your teeth loosen. Your immune system collapses. You die. That is scurvy. That is what the broken gulo gene can do to a human being if the food supply fails. British sailors in the 16th and 17th centuries died from this by the hundreds. Ships would lose half their crew on long voyages, not from storms, not from battle, from a gene that stopped working 40 million years ago. It took humanity centuries to figure out that citrus fruits were the solution, to figure out that we needed to get from outside what every other mammal gets from inside. So here is the question that should keep you up at night. If this gene is so critical, if its absence can kill you, why does every human carry the broken version? Why did natural selection not eliminate this mutation in 40 million years? Why are we all alive right now with a gene in our cells that could have killed each and every one of our ancestors? The answer to that question turns out to be the beginning of a much stranger story, because gulo is not the only broken gene in your genome. It is not even close to the only one. Your DNA is full of shattered sequences and deleted instructions and mutated machinery that, by every rule of evolutionary biology, should have been ruthlessly eliminated before it could spread through a population. And yet here it is. Here we are. And here is the part that makes no sense. Most of these errors, these broken and mutated and deleted pieces of genetic code, did not just fail to kill us, they built us. The human brain, which is the most structurally complex object ever discovered anywhere in the known universe, appears to have been assembled almost entirely from mistakes, accidental duplications, misfired copies, mutations that disrupted ancient proteins, deletions of sequences that have been doing the same job since before the dinosaurs. And somehow, through a chain of events that biologists are only now beginning to reconstruct, these accumulated errors produced language, art, mathematics, music, philosophy, and the ability to look up at the night sky and wonder what it is. That is the human paradox, the thing that makes no clean sense when you hold it against everything we know about how evolution is supposed to work. We are, by every reasonable measure, a collection of genetic accidents that survived when they should not have, persisted when they should have, been eliminated, compounded when they should have cancelled out, and the result is the only creature in 4 billion years of life on this planet that can sit with a question and not be satisfied until it has an answer. Nobody fully understands how this happened. The science is real, the evidence is extraordinary, but the complete picture is still being assembled piece by piece in laboratories around the world. And that is part of what makes this story so extraordinary. We are close enough to understanding to see the outline of the answer, far enough away that the mystery is still alive. So let's start at the beginning. Let's start with the machinery that was supposed to prevent all of this from happening. Because to understand why these errors survived, you first have to understand why they should not have. Natural selection is not a gentle process. That is not a metaphor, and it is not hyperbole. It is a description of something that has been operating on every living thing for 4 billion years, without pause, without mercy, and without any awareness of what it is doing or what it is building. It has no goal. It has no direction. It is not trying to create anything. It is only doing one thing, removing what does not work. Every time a cell divides, the DNA inside it is copied. 3 billion base pairs of genetic code duplicated from scratch every time. And every copy introduces errors. The cellular machinery that does this copying is extraordinarily accurate. It makes roughly 1 mistake per billion base pairs. That sounds almost perfect, but 1 per billion across 3 billion base pairs means about 2 to 3 new mutations every single time a human cell divides. Multiply that by the trillions of cell divisions that happen during a lifetime, and you're talking about an enormous number of mutations accumulating in a single body over a single life. Most of them do nothing. They land in parts of the genome that are not encoding anything critical. They change a single letter in a sequence that turns out to be redundant. They are silent passengers, carried along, doing neither harm nor good. Scientists call these neutral mutations. They are the background noise of the genome. But some mutations land in the wrong place. They hit a protein coding gene and change the shape of the protein it encodes. They delete a regulatory region that controls when a gene turns on during development. They disrupt an enzyme that has been functioning the same way since before multicellular life existed. These are the mutations that matter. These are the ones that change how an organism functions in the world. And when a mutation changes function in a direction that reduces survival or reproductive success, natural selection removes it. The individual carrying the mutation either dies before it can reproduce, or reproduces less successfully, or passes the mutation on at a reduced rate. Generation after generation, the bad mutation gets diluted. It spreads less and less until it is gone. This is purifying selection, the filter, the mechanism that keeps the genome clean, functional, and adapted. It works with extraordinary precision when you give it enough time. We know it works because we can see conserved sequences, stretches of DNA that have stayed essentially identical for 300 million years across wildly different species. A gene sequence that is identical in humans, mice, chickens, and fish has been sitting under purifying selection for the entire time those lineages has been diverging. Every mutation that tried to change it was eliminated. Only the functional version survived. That is how powerful the filter is when it is operating at full strength. But the filter is not perfect. There are specific, well-documented conditions under which genuinely harmful mutations can slip through. Conditions under which a mutation that should have been eliminated instead persists, spreads, and eventually becomes fixed in a population, meaning every individual carries it. These conditions matter enormously when you're trying to understand why the human genome looks the way it does. The first condition is called genetic drift. In small populations, random chance matters more than fitness. If a harmful mutation happens to appear in an individual who also happens to be in the right place at the right time, who survives a drought or an epidemic through sheer luck, rather than because of the mutation, that mutation gets passed on anyway. In large populations, this randomness averages out. The filter works. But in small populations, bottlenecks, founder effects, isolated groups, drift can fix even harmful mutations simply because the population is too small for selection to overcome the noise of chance. The second condition is the heterozygote advantage. Sometimes a mutation that is harmful when you have 2 copies is actually beneficial when you have only 1. The most famous example is sickle cell anemia. 2 copies of the sickle cell mutation causes serious disease, but 1 copy, while not causing disease, provides partial resistance to malaria. In regions where malaria is common, individuals with 1 copy survive better than individuals with 0 copies, so the mutation spreads and stays in the population even though it causes suffering when 2 copies are inherited. The third condition is pleiotropy. A gene rarely does only one thing. Most genes affect multiple traits, multiple pathways, multiple processes. A mutation that disrupts one function might, at the same time, accidentally enhance another. The net effect might be positive even if part of the effect is negative. Selection evaluates the total package, not the individual components. And the fourth condition, the most relevant one for understanding human evolution, is the changing environment. What is harmful in one environment can become neutral or even beneficial in a different one. When the environment changes, the calculus of survival changes. Mutations that were being filtered out suddenly face a different fitness landscape. And mutations that were tolerated as neutral can suddenly become advantageous. Now hold all of that in mind, because over the next 3 million years of human evolution, every one of these conditions was in play simultaneously. Small, isolated populations, rapid environmental change, new food sources, new climates, new social structures that rewrote the rules of survival. And in the middle of all that upheaval, a series of genetic mutations appeared that, under any ordinary circumstances, should have been disasters. They were not disasters, they were us. To understand what these mutations actually did, you first need to understand what DNA actually is. Not the simplified version, the real thing. Because the real thing is stranger and more beautiful than any simplification can capture, and the strangeness is important. DNA is a molecule, a very long, very specific molecule. In humans, if you took the DNA from a single cell and stretched it out into a straight line, it would be about 2 meters long. 2 meters of molecular code, coiled and folded and compacted into a nucleus that is about 5 micrometers across. That is roughly 100,000 times smaller than the width of a human hair. The engineering required to fit 2 meters of information into that space is something that no human technology has come close to replicating. That molecule is made of 4 chemical letters, 4 nucleotide bases: adenine, thymine, cytosine, and guanine, always abbreviated as A, T, C, and G. The human genome contains approximately 3 billion of these letters arranged in a specific sequence. 3 billion. If you printed the human genome in a standard font at a normal point size, the printout would be roughly 3,000 volumes the size of a standard encyclopedia. Every cell in your body, except red blood cells and platelets, contains the full 3,000 volume text. All of it, copied every time a cell divides. Most of that text, and this will surprise many people, does not encode proteins. Only about 1.5% of the human genome is what scientists call protein coding sequence. The rest was called junk DNA for a long time, and scientists now understand that this was a mistake. Much of the non-coding genome contains regulatory sequences, enhancers, silencers, promoters, scaffolding elements, sequences that control when and where and how strongly genes are expressed. The instructions for building a human being are not just in the genes. They are in the spaces between the genes, in the punctuation, not just the words. When a mutation happens in the protein coding sequence, the effect can be direct and dramatic. Change one letter in the middle of a gene, and you might change the shape of the protein it encodes. Change the shape of the protein, and you change its function. Change its function, and you change what the cell that produces it can do. This is the traditional view of mutation, the one that makes it into textbooks. But when a mutation happens in a regulatory region, the effect is different. Not necessarily smaller, sometimes larger. Because regulatory regions control expression, and expression controls everything. A regulatory mutation does not change the shape of a protein. It changes when, where, how much, and in what combination the protein is produced. Same protein, different timing, same protein, different location. And that difference in timing and location can transform development entirely. This distinction matters enormously for understanding human evolution, because when scientists actually examine the mutations that separate human DNA from chimpanzee DNA, most of the differences that seem to matter were not in the protein coding genes. Most of the interesting differences were in the regulatory regions, the volume controls, the switches, the timing circuits. Humans and chimpanzees share roughly 98.8% of their protein coding DNA. We are nearly identical at the level of individual proteins. What makes us different is not so much what proteins we have, but when they are produced, where they are produced, for how long, and in what sequence during development. The genome is less like a blueprint than it is like a symphony score. The notes are almost the same. What changed is the tempo, the dynamics, the orchestration. And what changed that orchestration was, in many cases, an error. A mutation that landed in a regulatory region and accidentally rewrote the timing of something critical. The way a wrong note played at the right moment in an improvisation can become the seed of an entirely new melody. Now we can start to talk about the errors themselves, the specific mutations, the ones that matter, the ones that built us from the inside out. In 2004, a geneticist named Hansel Stedman published a paper, in the journal Nature that triggered one of the more heated debates in paleoanthropology. The paper was titled, straightforwardly enough, "Myosin Gene Mutation Correlates with Anatomical Changes in the Human Lineage," and the argument it made was simple enough to state, but extraordinary in its implications. 2.5 million years ago, give or take, something happened to the jaw muscles of our ancestors. Not a trauma, not a disease, a mutation, a tiny deletion in a gene called MYH16. 16 base pairs removed from a sequence of code that had been essentially unchanged for millions of years before that. MYH16 encodes a specific type of myosin, which is the molecular motor protein responsible for muscle contraction. This particular myosin is expressed primarily in one muscle, the temporalis. The temporalis is the large muscle that runs from the side of your skull down to your jaw. It is one of the primary muscles responsible for closing the jaw and for the crushing force of your bite. Before the deletion, this muscle was enormous. In all other great apes, the temporalis is a massive structure that extends across most of the skull. In chimpanzees and gorillas, the temporalis muscle is so large that in males it actually meets at the top of the skull and requires a bony ridge, called a sagittal crest, to provide enough anchoring surface. You can see that crest as the raised ridge that runs along the top of a gorilla's head. It is there because the jaw muscles are so large and so powerful that the skull needs extra surface area to hold them. In humans, the temporalis muscle is much smaller. Our skulls are smooth and domed at the top, with no sagittal crest. Our jaws are weaker, relatively speaking, than the jaws of every other great ape. A chimpanzee can generate roughly 3 times the bite force of a human. For an animal that depends on its teeth for food processing and for defense, a mutation that drastically weakens the jaw muscles should be a serious disadvantage. It should be selected against. It should be removed from the population. But Stedman's team noticed something. When they mapped the timing of the MYH16 mutation using molecular clock estimates, they placed the deletion at approximately 2.5 million years ago. And 2.5 million years ago is one of the most important moments in human evolutionary history. It is precisely when the fossil record shows the first dramatic expansion of the hominid brain case, when the skulls of our ancestors suddenly start getting noticeably bigger. When the genus Homo appears, the hypothesis Stedman proposed was elegant and immediately controversial. The temporalis muscle, because of its size and attachment points across the skull, physically constrains the growth of the bones it anchors to. In great apes, the temporal and frontal bones of the skull cannot expand freely in certain directions, because the massive temporalis muscle is sutured to them. The muscle holds the skull in a particular shape. When the MYH16 mutation drastically reduced the size of the temporalis, that physical constraint was removed. The skull bones were free to expand in new directions, and under selective pressure to accommodate a larger brain, they did. Think about what that means. The mutation that weakened the jaw, the mutation that made our ancestors worse at one of the most basic survival tasks an animal faces, the one that reduced their ability to bite, to crush, to defend themselves with their teeth. That mutation may have been the key that unlocked the skull, the error that removed a structural constraint, the mistake that made room. The controversy came fast. Other researchers pointed out that correlation is not causation. Yes, the timing aligns. But the skull's growth could have been driven by many things, and the MYH16 mutation might have been a passenger along for the ride rather than a driver of change. The relaxed jaw muscles might have been a consequence of dietary changes that were themselves driven by other adaptations, rather than a trigger for brain expansion. There was also a more fundamental objection. Natural selection would not have favoured a weaker jaw unless something else was already compensating. Our ancestors could not simply exist with weaker jaws unless their diet was changing at the same time, unless they were processing food in new ways, using fire or stone tools to do the work that their teeth could no longer do. And the earliest stone tools appear in the fossil record right around the same time as the MYH16 mutation, 2.5 million years ago, the same window. So maybe it was all happening simultaneously and interactively. Maybe weaker jaws and bigger brains and stone tools were all part of the same cascade of changes, each one enabling the others, each one creating conditions under which the others could be selected for rather than against. A mutation that weakened the jaw would have been catastrophic in an animal that had no other way to process food. But in an animal that was already using tools to break down tough plant material, in an animal whose hands were increasingly nimble and whose social group was increasingly sophisticated, the weaker jaw was survivable. And if that weaker jaw also, even slightly, even indirectly, enabled more brain growth, then the trade-off made evolutionary sense. What haunts me about this is how narrow the window is, how precisely things had to be aligned. The MYH16 mutation had to appear at a moment when the population was already shifting its diet, when tools were already in use, when the brain was already under selection pressure to expand. A few 100,000 years earlier, when the diet was different, when there were no tools, a mutation that halved your bite force would simply have killed you. The error would have been eliminated. The door would never have opened. But timing is everything in evolution. And 2.5 million years ago, the timing was just right. And that was only the first error. There are others coming. Bigger ones, stranger ones. Errors that did not just remove constraints, but actively, almost intentionally, rewired the way the brain developed. Errors that, if you did not know their origin, you might mistake for deliberate engineering. If the MYH16 mutation was the error that opened the door, then the next error was the one that built the room behind it. It is called SGAP2C. The name is a mouthful, and the story behind it takes a moment to set up, but stay with me, because this is one of the strangest and most important pieces of the human genome story, and the strangeness is not in the name. The strangeness is in what it does. Your brain contains somewhere between 80 and 100 billion neurons. Each neuron is a cell specialized for transmitting electrical signals. But neurons do not transmit signals to each other through direct physical contact the way copper wires conduct electricity. They communicate across tiny gaps called synapses. At each synapse, one neuron releases chemical messengers, neurotransmitters, which float across the gap and bind to receptors on the receiving neuron, triggering or suppressing a new electrical signal. Each neuron typically forms thousands of synaptic connections with neighboring neurons, and the pattern of those connections, the precise wiring diagram of which neuron is connected to which and how strongly, is what we call the connectome. It is the physical substrate of thought, memory, perception, and consciousness. The formation of those synaptic connections does not happen all at once. It happens gradually over years of development, and it is controlled by a complex toolkit of molecular signals that tell neurons when to reach out, when to form a connection, when to strengthen it, and critically, when to prune it back. Because synapse formation is not just a matter of building more connections, the mature brain is also defined by what gets removed. Neurons that fire together wire together, and neurons that do not fire together lose their connections. The brain refines itself through use, through experience, through the selective strengthening and pruning of connections in response to learning. The timing of this process matters enormously. In most mammals, synaptic development happens relatively quickly after birth. The critical period, the window during which the brain is most plastic, most responsive to experience, most capable of rewiring itself in response to input, is relatively short. A mouse's critical period for visual development lasts a few weeks. A cat's lasts a few months. Then the connections stabilize, the plasticity decreases, and the adult brain becomes less capable of dramatic rewiring. In humans, the critical period is extraordinarily long. The human brain is still undergoing significant synaptic pruning and reorganization well into the mid-20s. The window of plasticity, the period of maximum learning capacity, of maximum sensitivity to experience and environment, stretches across 2.5 decades of life. This is deeply unusual in the animal kingdom. Most mammals reach neurological maturity much faster. The elongated human critical period is one of the things that makes human childhood both so dependent and so extraordinary. We spend so long as helpless infants, and then as learning children precisely because our brains are still being actively built during that whole time. Now here is the mutation. The original version of the SRGAP2 gene, the ancestral version, is called SRGAP2A. It encodes a protein that is involved in the regulation of synaptic development. Specifically, it limits the density of dendritic spines, the tiny projections on neurons where synapses form, and it helps drive the maturation of those spines toward a stable adult state. About 3.5 million years ago, in the genome of some early hominin ancestor, a portion of the SRGAP2A gene was accidentally duplicated. Not the whole gene, a piece of it. A partial copy appeared in a different location on a different chromosome. This copy is called SRGAP2B. It was probably mostly functionless at first, a redundant fragment sitting in the genome doing nothing in particular. Then, roughly 2.5 million years ago, the same piece was accidentally duplicated again. Another partial copy. This one is called SRGAP2C. And SRGAP2C turned out to be not just a copy of part of SRGAP2A. Because of how it was truncated, because of the specific region that was duplicated and the specific region that was left out, SRGAP2C encodes a protein that binds to the original SRGAP2A protein and partially blocks it. SRGAP2C is an inhibitor of its parent gene, a competitive antagonist, a molecular jamming signal. When SRGAP2C interferes with SRGAP2A, the effect on synaptic development is to slow it down. Dendritic spine maturation is delayed. The spines that form are denser than they would otherwise be, and the process of stabilization, the shift from a plastic, rapidly changing synaptic state to a stable adult state, takes longer. In other words, SRGAP2C made our brains develop more slowly. And that should be bad. Slower development means a longer period of vulnerability, a longer period of dependence, a longer period during which the organism is not fully functional, not fully capable of surviving on its own. in almost any other animal, a mutation that prolonged the developmental period, that delayed neurological maturity, that extended the period of synaptic plasticity, would be a serious disadvantage. It would mean a longer time as prey, a longer time requiring parental investment, a longer time before the animal could be reproductively independent. But in humans, with our social groups, with our tool use, with our increasingly complex cooperative structures, the calculation changed. In humans, a longer critical period was not a vulnerability, it was a capacity. A longer window of plasticity meant a longer window during which the brain could be shaped by experience, by language, by culture, by learning that could not be encoded in the genome itself but could be transmitted socially, from adult to child over the decades-long span of human childhood. And the denser dendritic spines meant more synaptic connections per neuron. More connections mean more computational capacity. More connections mean more pathways for associations, for pattern recognition, for the kind of analogical thinking that underlies complex cognition. The brains of humans actually have more immature-looking dendritic spines, as measured against equivalent-age chimpanzee neurons well into adulthood. We are, in a very specific neurological sense, permanently slightly childlike, permanently in a state of higher plasticity than our nearest relatives. This property has a name in developmental biology. It is called neoteny, the retention of juvenile characteristics into adulthood, and it is one of the most striking features of the human species when you look at us from a developmental biology perspective. Our brains retain features of the developing brain. Our bodies, compared to chimpanzees, are more slender and upright and proportionally large-headed. Even our social behavior, our playfulness, our curiosity, our openness to new experience, has features that in other primates you see mostly in juveniles. sRGAP2C appears to be a key part of why. Not the only part, not even necessarily the biggest part, but a real, measurable, functional part. A mutation that slowed down brain development and accidentally created the foundation for human learning. This was published in 2012 by a team led by Céline Charrier and Evan Eichler. The paper appeared in the journal Cell, and it immediately raised the kind of question that makes you sit back and stare at the ceiling for a while. Not because the science is wrong, the science is solid, but because of what it implies. Evolution did not design human intelligence. There was no foresight, no planning, no intent. A partial gene duplication happened by accident 2.5 million years ago. The resulting protein fragment happened to partially interfere with a synaptic maturation gene. The resulting effect happened to slow brain development during a period when slower development was, for the first time in evolutionary history, actually useful. And that accident compounded with the other accidents, with the jaw muscle mutation, and the changing diet, and the social structures and the tools, and together they pushed a lineage in a direction that no evolutionary process had ever gone before. But here is the thing that really gets me. The timing. sRGAP2C appears roughly 2.5 million years ago. MYH16 mutates roughly 2.5 million years ago. The first Homo fossils appear roughly 2.5 million years ago. The first stone tools appear roughly 2.5 million years ago. The same window. The same narrow slice of time in which multiple independent mutations, multiple independent changes, all happened simultaneously, all in the same direction, all compounding each other. Either that is an extraordinary coincidence, or it is telling us something about the conditions that existed 2.5 million years ago. about how fast things were changing, about how much the selection pressure had shifted, about how many different changes could be simultaneously advantageous in a world that was rewarding intelligence above almost everything else. And it was not over, not even close. If you're using this to fall asleep tonight, that is exactly why I make these videos. Maybe subscribe and let me know where you're watching from and what time it is. I always check the comments, and it is fascinating to see people from all over the world settling in at completely different hours. Anyway, back to the genome, because we have only just started. There is a category of genomic sequence that scientists call conserved non-coding elements. These are regions of the genome that do not encode proteins. They sit in the non-coding portions of the DNA, and they are identical or nearly identical across dozens or hundreds of vertebrate species, identical or nearly identical between fish and frogs and lizards and birds and mice and elephants and humans. When scientists find a stretch of sequence that has stayed essentially unchanged for 300 million years across animals as different as a zebrafish and a chicken, they know that sequence is under extremely strong purifying selection. It is being preserved because any mutation that changes it causes enough harm to be eliminated. The sequence is functional, critical. The body needs it exactly as it is. In 2006, a team led by Catherine Pollard at the University of California published a paper in Nature that identified something remarkable. They were comparing the human genome to the chimpanzee genome and to the genomes of other vertebrates, and they were specifically looking for conserved non-coding elements, sequences that had been stable for hundreds of millions of years of vertebrate evolution, and they found 2,649 of them, conserved for hundreds of millions of years, the same in fish and frog and chicken and mouse, almost identical across the entire vertebrate phylogeny. But in the human lineage, these sequences had changed, not gradually, not slowly, not at the background rate of neutral mutation. They had changed rapidly, dramatically, way faster than the neutral rate, way faster than any other part of the genome, and way faster than the same sequences had changed in any other vertebrate lineage. These regions are called human accelerated regions, HARs, and the very existence of them is a paradox. If a sequence has been conserved for 300 million years under strong purifying selection, every mutation that tried to change it was eliminated. The sequence is under lock and key. It should not change. It cannot change without causing harm. And yet in the human lineage specifically it changed, repeatedly, rapidly. There are only two explanations. Either the sequence stopped being functionally important in the human lineage, and neutral mutations accumulated because the purifying selection was relaxed, or the sequence started being positively selected for change, not just tolerated, actively selected. Mutations that changed these sequences increased fitness. Individuals who carried the changed versions survived and reproduced better than individuals who carried the original versions, and so the changes spread through the human lineage rapidly, driven not by drift, but by selection. The second explanation is the one the data supports. The evidence points to positive selection. The human lineage actively selected for changes in regions that had been locked down for hundreds of millions of years in every other vertebrate. Why? What was being selected for? The most famous human accelerated region is called HR1, When Pollard's team looked at HSR1, they found that it had changed at a rate 50 times faster than the neutral mutation rate in the human lineage. In the 300 million years of vertebrate evolution before hominins, HAR1 accumulated a total of 2 changes. In the roughly 6 million years since the human and chimpanzee lineages diverged, HAR1 accumulated 18 changes. 18 changes in 6 million years, where there had been only 2 changes in 300 million years. The region did not just change, it exploded. What does HAR1 do? This is where it gets strange in a way that feels almost too on the nose. HAR1 is part of a regulatory RNA molecule, not a protein-coding gene, a regulatory RNA, which means its function is to control the expression of other genes, and specifically HAR1 is expressed in a specific type of neuron, Cajal-Retzius neurons, during a specific window of fetal brain development, between weeks 7 and 19 of gestation, during exactly the period when the cortex is being laid down, when the fundamental architecture of the cerebral cortex is being established. The cortex is the most sophisticated structure in the human brain. It is where conscious thought happens, where language is processed, where abstract reasoning occurs, where memories are formed and retrieved. The architecture of the cortex, how many layers it has, how it is organized, how it folds and expands is central to everything that makes human cognition unusual. And HAR1 helps regulate cortical development during the critical window. The human version of HAR1 is different from the version in every other vertebrate. It changed faster in the human lineage than almost anything else in the genome, and it appears to be involved in building the most distinctively human part of the brain. There are another 2,648 human accelerated regions. Some have been studied, many have not. Most of them, like HAR1, are in regulatory regions rather than protein coding sequences. They appear to control the expression of genes involved in neural development, in cognitive function, in the timing and architecture of brain construction. They changed rapidly in the human lineage specifically, and they suggest that the evolution of human cognition was not primarily about acquiring new genes. It was about rewiring the regulatory network that controls how existing genes are used, changing the symphony by changing the score, not by changing the instruments. This is a deeply uncomfortable idea for a certain kind of intuitive thinking about evolution. We want the story to be about new genes emerging and doing new things. But the HARs tell a different story. They tell a story about ancient sequences that had been locked in place for hundreds of millions of years suddenly being unlocked in one lineage, about regulatory circuits that had been frozen suddenly being rewritten, about the timing and architecture of development being restructured rapidly and specifically in the lineage that led to us. And nobody fully understands why the lock opened, why the purifying selection that had held these sequences stable for hundreds of millions of years was suddenly relaxed or reversed specifically in the hominin lineage, why the selection pressure shifted so dramatically that changes in these regulatory regions suddenly became advantageous rather than harmful. That question does not have a clean answer, and that is part of what makes the human accelerated regions one of the most fascinating and unresolved stories in all of genomics. By 2015, geneticists had a new tool that they had not had before. They could sequence the genomes of ancient hominins, Neanderthals, Denisovans, early modern humans, and compare those ancient sequences to modern humans and to chimpanzees. They could map mutations with a precision that earlier studies could only approximate. And two groups working independently on different continents found something extraordinary in 2018. They were both looking at a specific region of chromosome 1. Both groups identified the same thing, a gene, or rather a set of gene duplications, that appeared to be unique to modern humans and closely related to the emergence of the large human brain. The gene is called NOTCH2NL. NOTCH2 is an ancient gene. It has been present in vertebrates for hundreds of millions of years. It is a critical component of the Notch signaling pathway, which is involved in controlling the behavior of stem cells throughout the body, including neural stem cells, the cells that produce neurons during brain development. It is one of the most fundamental and conserved regulatory genes in animal biology. About 3 to 4 million years ago, in the hominin lineage, part of the Notch2 gene was duplicated. Not the whole gene, a portion of it, specifically a portion of the extracellular domain, the part of the protein that receives signals from other cells. These partial duplicates, the Notch2NL genes, encode truncated versions of the Notch2 protein, and because they are truncated versions, they can interfere with the normal signaling function of the full-length Notch2. They compete for the same binding sites. They dilute the signal. The effect of this interference on neural progenitor cells, the cells that give rise to neurons during cortical development, is to delay their differentiation. Normally, neural progenitor cells go through a certain number of division cycles and then exit the cell cycle and become neurons. The Notch signaling pathway is one of the systems that controls when this happens, when the progenitor cells stop dividing and start differentiating. When Notch2NL interferes with normal Notch2 signaling, the progenitor cells stay in the dividing state for longer. They produce more cells before differentiating. And more progenitor cells means more neurons. More neurons means a larger cortex. More cortex means more computational capacity. The human cortex is substantially larger, relative to body size, than the cortex of any other animal. The cortex of a human brain is not just bigger than a chimpanzee's cortex, it is more folded. The surface area is dramatically expanded by a complex system of gyri and sulci, ridges and grooves that pack a much larger surface area into a confined skull. This folding is what you are seeing when you look at a picture of a human brain. That wrinkled, convoluted surface is not random. It is the result of a cortex that has grown so large that it has to fold to fit inside the skull. The NOTCH2NL duplications appear to be part of what drove that expansion. Fides and his colleagues modelled it and estimated that the NOTCH2NL genes could account for roughly a third of the increase in cortical neuron number that separates humans from other great apes. That is an extraordinary fraction for a single genetic change, or a small cluster of related changes. But here is the dark side, because there is always a dark side. The chromosomal region where NOTCH2NL sits is what geneticists call a copy number variable region. The number of copies of the NOTCH2NL genes is not fixed. It can vary from person to person. Most people carry a certain number of copies, but some people carry more and some carry fewer, and when the copy number deviates from the typical range, things go wrong. People with extra copies of the NOTCH2NL region are more likely to have macrocephaly, an unusually large head circumference, and they are significantly more likely to have autism spectrum disorder or other neurodevelopmental conditions. People with too few copies are more likely to have microcephaly, an unusually small brain, and they are more likely to have schizophrenia. More of the brain expansion gene. Brain grows too large, connectivity becomes atypical, autism. Less of the brain expansion gene. Brain grows too small, connectivity becomes dysregulated, schizophrenia. The same gene whose duplications appear to have expanded the human cortex is also, in altered dosage, a major risk factor for two of the most common and most debilitating neurological conditions in the human population. That is not a coincidence. That is a reflection of something deep about how human intelligence was built. It was built at the edge of something, at a threshold where more of the same is sometimes too much, and less of the same is sometimes too little, and the narrow range in the middle is where function lives. And that range is narrow precisely because the brain expansion was pushed so far, so fast. When you optimize for maximum cognitive capacity, you are optimizing in a region where the margin for error is small. And small margins mean that random variation in gene copy number, which happens all the time, can push you outside the functional range. Human intelligence is balanced on a knife edge. The same genetic architecture that enables it also makes it fragile. There is another brain expansion gene. It is called ARHGAP11B, and it is even more directly connected to the folding and expansion of the cerebral cortex than Notch2NL. The original gene, ARHGAP11A, is present in all vertebrates and encodes a protein involved in cell division. About 3.5 million years ago, a portion of ARHGAP11A was duplicated in the hominin lineage. The duplicate ARHGAP11B has a specific mutation, a single base change that altered the reading frame of part of the gene. That single letter change transformed part of what would have been a non-functional redundant copy into a new sequence that encodes a functional protein with a new property. ARHGAP11B is expressed in basal radial glia, a specific type of neural progenitor cell that is particularly abundant in humans compared to other primates. These cells are found in what is called the outer subventricular zone of the developing cortex, and their abundance in the human lineage is directly correlated with the size and complexity of the cortex. More basal radial glia means more neurons, means more cortex, means more folding. In 2015, Madeleine Florio and her colleagues at the Max Planck Institute in Dresden showed something remarkable. They took the ARHGAP11B gene from humans and inserted it into mice. Mice do not normally have ARHGAP11B. They have only the ancestral ARHGAP11A. When the researchers introduced the human version, the mouse brains enlarged. The cortex expanded. The normally smooth mouse cortex began to develop folds, gyri, the kind of sulcal pattern that is characteristic of human brains and is almost entirely absent in mice. They had inserted a human gene into a mouse and gotten a more human-looking brain, just from that one gene, just from ARHGAP11B. Later experiments with ferrets and non-human primates confirm the effect. ARHGAP11B, when introduced into a developing brain, drives cortical expansion. When you look at the timeline, ARHGAP11B appears in the hominin lineage around 3.5 million years ago. The cortex of hominins begins expanding dramatically around 2.5 to 3 million years ago. The gene is present in Neanderthal and Denisovan genomes, which means our cousins, who also had enlarged brains, also had this gene. It is absent in chimpanzees and gorillas, which means the gene is specific to the group of hominins that evolved large brains. The evidence is as strong as it gets in ancient genetics. A RHGAP11B did not just appear alongside brain expansion, it appears to have driven it. A partial gene duplication, a single base change that altered the reading frame, and the result was a new functional gene that expanded the cortex of every hominin lineage that carried it. From a single error in the copying of an ancient gene 3.5 million years ago, in a population of early hominins living on an African savanna who would have had no idea anything unusual had happened. That error propagated forward through every generation. It expressed itself in the developing brain of every individual who carried it, expanding their cortex slightly, making their neural architecture slightly more complex. And over millions of years it accumulated alongside the other errors, the jaw muscle mutation and the synaptic development slowdown, and the HAR changes and the NOTCH2-male duplications, each one building on the others, each one making sense only in combination with the others. And 2.5 million years later, the cumulative product of all these errors looked up at the African sky and started trying to understand what the stars were. Let's step away from the genetics for a moment. Let's think about what it actually means to live inside a brain that was built this way. Not the theory, not the molecular biology, the lived reality. What it feels like from the inside to be the product of accumulated errors that nobody designed and nobody intended. Start with something simple. Start with the fact that human infants are the most helpless newborns of any large mammal on Earth. A foal can stand within hours of birth. A chimp infant can cling to its mother and support its own weight within days. A human infant cannot hold up its own head. It cannot regulate its own body temperature. It cannot feed itself, orient itself, protect itself from anything. For months, it is almost entirely dependent on other members of its species for survival. Why? Because the human brain is too big to be born fully developed. The skull of a full-term human fetus is already close to the maximum size that can pass through the birth canal. If the brain were allowed to develop further before birth, if humans tried to give birth to offspring whose brains were already at a more advanced stage of development, mothers would not survive the birth. The birth canal would be too narrow, the head would be too large. So human infants are born neurologically premature. Their brains are still in an early stage of development at birth. They are approximately 40% of the way through the growth they will ultimately achieve. They emerge helpless because they have to, because the alternative, staying in the womb until the brain was more developed, is not physiologically compatible with human anatomy. This is called the obstetric dilemma. The human pelvis evolved for bipedal locomotion, for walking upright, which requires a different pelvic geometry than the ape pelvis. But a human-sized brain requires a large head that the pelvis has to accommodate during birth. These two demands are in direct conflict. The pelvis cannot be both optimally shaped for walking and optimally shaped for birth. The compromise is a pelvis that works for walking, but only barely accommodates birth at the cost of birthing neurologically immature offspring. This is not a minor inconvenience. Childbirth in humans has historically been dangerous for both mother and infant in a way that is essentially without parallel in the animal kingdom. Human birth rates of complication and death were, before modern medicine, devastatingly high. Human societies developed elaborate systems of midwifery, birth rituals, and social support for labouring women, precisely because birth in our species is uniquely difficult and uniquely in need of assistance. We evolved a form of cooperative birth, birth attended by helpers, as a direct consequence of the obstetric dilemma. The obstetric dilemma is a direct consequence of the brain expansion genes of ARHGAP11B and NOTCH2NL, and the other duplications and regulatory changes that expanded the cortex. More brain means bigger head at birth, means more dangerous birth. This is the cost that the brain expansion genes imposed, not on distant ancestors, on every woman who has ever given birth, on every culture that has ever had to figure out how to support the most dangerous thing a human body can do, and then consider what happens after birth. The human infant is born with a brain that is, in a very real sense, still under construction. The critical period of plasticity that SRGAP2C helped extend begins at birth and does not end for decades. During that entire time, the brain is being wired by experience, by language, by what it sees and hears and feels and practices. The neural circuits for reading, which no human brain comes pre-wired to do, are assembled through years of instruction and practice, borrowing and repurposing circuits that originally evolved for visual processing and speech processing. The neural circuits for mathematics are assembled through explicit teaching. The cultural knowledge, the historical memory, the technical skills that allow a human adult to function in a complex society, none of it is in the genome. All of it is inscribed laboriously through years of learning into a brain that is designed to be inscribed. This is why childhood lasts so long in humans. Not as a social convention, not as a cultural invention, as a biological necessity. The human brain requires roughly 25 years of experience-dependent development before it reaches full maturity. The prefrontal cortex, the region most associated with planning, impulse control, long-term thinking and complex decision-making, does not finish developing until the mid-20s. This is why adolescents make decisions that seem, from an adult perspective, to underweigh future consequences. It is not immaturity in a purely social sense, it is neurological reality. The decision-making circuits are literally still under construction. SRGAP2C slowed the timeline. The brain expansion genes made the brain bigger. The HARs rewired the regulatory circuits, and the result is a developmental program that stretches across a quarter of a century, during which the organism is profoundly dependent on its social group for survival, learning, and the accumulation of the knowledge that the genome cannot encode. This is simultaneously the most extraordinary and the most expensive strategy in the history of biology. No other animal invests this much time and resources per offspring. No other animal requires this many years of post-birth brain development. No other animal depends so thoroughly on cultural transmission of knowledge, on the explicit teaching of each generation by the previous one, for the skills it needs to survive. Think about what a human being actually is, stripped of all cultural scaffolding. Without the accumulated knowledge of your culture, without language, without the years of teaching you received, you are not merely a less educated human. You are an animal that cannot start a reliable fire, cannot identify which plants are edible and which are toxic, cannot predict the behavior of the large predators in your environment, cannot build the kind of shelter you need to survive winter at the latitudes where many human populations live. You're dependent not just on your individual genome, but on the accumulated cultural knowledge of your group in a way that no other animal comes close to matching. That dependency, that extraordinary vulnerability, is the direct consequence of the brain expansion. The bigger brain required more developmental time. More developmental time required longer childhood. Longer childhood required more social support. More social support created the conditions in which cultural knowledge could accumulate across generations, each generation building on the last because there was always enough overlap between generations for teaching to happen. And once cultural accumulation began, the selection pressure for more brain, for more plasticity, for a longer critical period intensified. The process fed on itself. But there is another dimension to the human element, and it is harder to talk about, not because the science is uncertain, but because it touches something intimate. The same features of the genome that built the human brain also created the conditions for human mental suffering. The NOTCH 2NL copy number variation that expands the cortex in one direction causes autism when it goes too far and schizophrenia when it goes too short. The extraordinary synaptic density that SRGAP2C created, the denser, slower maturing spines, the extended critical period, these features make the human brain extraordinarily capable of learning and also extraordinarily sensitive to trauma. A brain that can be shaped by years of rich experience can also be deformed by years of chronic stress. The plasticity that makes human learning possible is the same plasticity that makes human trauma enduring. The same circuit-building capacity that allows a child to learn language and music and mathematics allows a child exposed to chronic abuse to build neural circuits tuned for threat, for hypervigilance, for the emotional patterns that persist long after the threat is gone. Human beings have the richest and most complex inner lives of any animal we know of. We also have rates of depression, anxiety, post-traumatic stress, psychosis, and cognitive disorder that are staggeringly high. These are not separate facts. They are the same fact viewed from two different angles. A brain capable of extraordinary depth and complexity is also a brain capable of extraordinary depth and complexity of suffering. The same architecture that enables one also enables the other. You cannot have the ceiling without accepting the floor. This is not a counsel of despair. It is an invitation to a specific kind of understanding. When you look at human mental suffering not as a malfunction to be ashamed of, but as the shadow cast by the architecture that also casts the light, something changes. The vulnerability is not separate from the intelligence. It is part of the same structure, built by the same errors, expressing the same fundamental property of a brain that was pushed further than any brain before it had gone. Now let's talk about the people we almost were, the other humans, the ones who walked alongside us, who shared campfires with some of our ancestors, who left their mark in our DNA in a way that we are still discovering. For most of human history we assumed we were alone, the only human species, the singular endpoint of an evolutionary process that had culminated in us. Other hominins were things of the past, relatives we had replaced, a story we had concluded. Then, in 2010, Svante Paabo and his team at the Max Planck Institute published something that rewrote the story. They had sequenced the Neanderthal genome, and when they compared it to modern human genomes, they found a signal that was not supposed to be there. Modern humans, specifically non-African modern humans, Europeans, Asians, indigenous Australians, indigenous Americans, carry between 1 and 4% Neanderthal DNA in their genomes. Not all non-Africans carry the same Neanderthal sequences. Different populations carry different pieces. But every non-African modern human carries some. And the presence of that DNA is not an artifact or a contamination. It is a real signal of interbreeding, of populations that were classified as different species actually mating and producing offspring with viable genomes, offspring who survived and reproduced and passed their ancestry forward to us. Our ancestors encountered Neanderthals as they expanded out of Africa, roughly 60 to 80,000 years ago. They met in the Middle East, in Europe, possibly in Central Asia. They interacted, and at least some of those interactions resulted in children who survived and integrated into one community or another. 1 to 4% sounds small. It is not. Not when you consider that it has been persisting in human populations for 60 to 80,000 years, which means it was not being removed by selection. If Neanderthal DNA were consistently harmful, it would have been eliminated over that many generations. The fact that it persists means it has either been neutral or, in at least some cases, beneficial, and some of it has been actively beneficial. Some Neanderthal-derived sequences are present at notably higher frequencies than you would expect from neutral drift. They are found in the genomes of specific populations at frequencies suggesting positive selection. The immune genes are a good example. The Neanderthals and Denisovans, both close relatives of ours, had been living in Eurasia for hundreds of thousands of years before modern humans arrived. They had been adapting their immune systems to the pathogens of Eurasian environments during that time. When modern humans arrived with immune systems tuned to African pathogens and encountered Eurasian diseases they had never been exposed to, some of the Neanderthal immune gene variants they introgressed turned out to provide critical protection. Several genes in the innate immune system, including genes that recognize and respond to viral infections, are present in modern human populations at frequencies that suggest they were positively selected after introgression from Neanderthals. We borrowed immune defenses from the people we were replacing. We carried forward their adaptations even as we carried forward our own. There are Neanderthal-derived variants in modern human populations that affect skin color, hair texture, and possibly adaptation to cold climates. The Neanderthals had been in cold northern latitudes for hundreds of thousands of years. They had evolved physiological adaptations to those environments. When modern humans moved into the same environments, some of the genetic tools the Neanderthals had developed were immediately useful. But the Neanderthal legacy is not all beneficial. There are Neanderthal-derived sequences in modern human genomes that are associated with increased risk of depression and anxiety. There are variants associated with increased susceptibility to blood clotting disorders. There are variants associated with increased pain sensitivity, with altered circadian rhythm, with higher likelihood of developing certain addictive behaviors. These are not large effects, they are probabilistic associations. Having the Neanderthal variant of a gene does not mean you will definitely develop depression or a clotting disorder. It means your risk is modestly elevated. But the presence of these variants raises a question. Why did they persist? If they increase risk of depression and anxiety, why were they not eliminated over 60,000 years? The most likely answer is context dependence. A gene variant that increases anxiety in a modern, stable, resource-rich environment might have been advantageous in the environment where it evolved. Heightened anxiety can make you more vigilant, more alert to threats, more likely to notice the ambiguous sound in the bushes and treat it as danger rather than noise. In an environment with real predators and real threats of starvation and real intergroup violence, that vigilance could be the difference between surviving and not. The variant was not a flaw in the environment it evolved in. It was an adaptation. It became a vulnerability only when the environment changed faster than selection could respond. This is a pattern that shows up repeatedly in the human genome. Adaptations that made sense in one context persisting into a context where they produce suffering. Not because the genome is broken, because the genome adapted to a world that no longer exists in the form it adapted to. And here is the stranger version of the Neanderthal story, the one that complicates the narrative of modern humans simply replacing their cousins. We did not just replace the Neanderthals, we absorbed them, partially, imperfectly. A few % of our genome carries their legacy. Their immune adaptations help keep us alive in environments their own species could no longer survive in. Their cold climate adaptations may have helped some of our ancestors push further north than they could have otherwise, and some of their cognitive architecture, whatever portion of their intelligence was encoded in gene sequences we introgressed, is inside us too. We are not pure, we never were. We are a mosaic, an accidental hybrid, carrying pieces of multiple lineages, having become something that none of those lineages was individually. The errors of our genome include borrowed errors that were not ours originally, that we picked up from people we met and sometimes displaced and sometimes became. If you are settling in now, finding a comfortable position, letting the day fall away, that is exactly right. Let the story keep unfolding. Let your mind hold it loosely. You do not need to keep track of every detail. Just let it wash over you. We have more to cover, but there is no rush. Let's talk about something that seems completely unrelated to brain evolution, but that is in fact part of the same story. Something that is visible on the outside of every human body rather than hidden inside the genome. Skin color. The ancestral human population that emerged in Africa had dark skin, highly melanized skin. Melanin is the pigment that gives skin its color, and it also functions as a barrier against ultraviolet radiation from the sun. Ultraviolet radiation is harmful to DNA. It causes mutations. It causes skin cancer. In high UV environments like equatorial Africa, high melanin levels are protective and are strongly selected for. When populations of modern humans moved out of Africa into higher latitudes, the UV environment changed. In Northern Europe, in Scandinavia, in the British Isles, the sun angle is lower and the UV intensity is substantially reduced. And UV radiation, the same UV radiation that damages DNA and causes cancer, is also the trigger for vitamin D synthesis in the skin. Vitamin D is essential. It is required for calcium absorption, for immune function, for neural development, for bone formation. Without adequate vitamin D, you develop rickets, immune dysfunction, increased susceptibility to infectious disease, and a range of developmental problems. In high UV equatorial environments, the sun provides enough UV even through darkly pigmented skin to generate adequate vitamin D. In low UV northern environments, darkly pigmented skin blocks too much of the limited UV available, and vitamin D synthesis becomes inadequate. So populations that moved to high latitudes faced a new selective pressure. Not for more brain capacity, not for social complexity, but for a simple metabolic issue. They needed to make vitamin D in their skin. And the way evolution addressed this, in population after population that moved to high latitudes, was to select for reduced melanin levels, lighter skin, less UV protection, less cancer protection, but more vitamin D production in a low UV environment. The specific genetic changes that produced light skin in European populations are now well characterized. There are several genes involved: SLC24A5, SLC45A2, HERC2, OCA2. Each has variants that reduce melanin production, and each was selected strongly in the European lineage after the migration out of Africa. The selection coefficient, the rate at which the lighter skin variants spread through the population, was very high, suggesting that vitamin D deficiency was causing serious fitness consequences in those early northern populations. But here is the error part of the story. These same populations carry the broken gulo gene from 40 million years ago. They cannot synthesize their own vitamin C. And now they were also in environments where they could not synthesize adequate vitamin D either, because the genetic toolkit they brought with them was adapted for an equatorial UV environment and not a northern one. They were, in a real sense, metabolically stranded in a new environment, dependent on dietary sources of both vitamin C and vitamin D that the environment of equatorial Africa had provided abundantly, but that the northern latitudes provided less reliably. The lighter skin solved the vitamin D problem, but it created a new one. In high UV environments, people with reduced melanin are at dramatically higher risk of skin cancer. UV radiation that would have been blocked by dark skin now penetrates to the DNA in the basal layer of the epidermis and causes the kind of mutation that leads to melanoma and other skin cancers. The populations that moved to high latitude solved one problem, vitamin D deficiency, by creating another cancer risk in high UV environments. This is not a failure of evolution. It is exactly what you would expect from a process that does not plan, does not optimize globally, and does not see around corners. In the low UV northern environment where the selection happened, the cancer risk was low enough to be tolerable, and the vitamin D benefit was substantial, enough to be strongly selected. The compromise made sense in the context where it was made. The problem only becomes visible from a global perspective, from the perspective of a population that has since moved, that now lives in high UV environments with a light skin that was optimized for low UV environments. The mismatch between the genome and the current environment, between the error's solution and the current problem, is a recurring theme in human biology. The genome adapts to specific environments. When the environment changes faster than the genome can follow, vulnerabilities emerge that were not vulnerabilities in the context where the adaptation occurred. Your genome was built for a world that no longer fully exists. That is not a special property of your genome. It is a property of every genome. The world always changes. The genome always lags. The only question is how large the lag is and what the lag costs. Here is another borrowed adaptation. This one comes not from Neanderthals, but from a cousin species we call the Denisovans. The Denisovans are known primarily from a few bone fragments found in Denisova Cave in Siberia. Their genome has been sequenced remarkably from the ancient DNA preserved in those fragments, and the Denisovan genome shows that they were a distinct population from both Neanderthals and modern humans, living in Asia during the time that Neanderthals dominated Europe. Modern humans who moved into Asia and the Pacific interbred with Denisovans, just as they had interbred with Neanderthals in the Middle East and Europe. And some of the Denisovan genetic sequences that entered modern human populations have been maintained and even amplified by positive selection. The most dramatic example is a gene called EPAS1. It encodes a protein called HIF2 alpha, which is part of the cellular oxygen sensing pathway. HIF2 alpha controls how cells respond to low oxygen levels. In most people, the cellular response to hypoxia involves increasing the production of red blood cells, which increases the oxygen carrying capacity of the blood. This is why people who move to high altitude, say, to live above 4,000 meters in the Himalayas or the Andes, initially feel the effects of oxygen deprivation. Their bodies need to adapt. Tibetans have lived at high altitudes for thousands of years and have adapted to do so without the chronic elevation of red blood cell levels that other populations experience at altitude. The chronic elevation of red blood cell levels that non-adapted people develop is actually harmful over long periods. It thickens the blood. It increases the risk of blood clots and stroke. It puts strain on the heart. Tibetans have a variant of the EPAS1 gene that allows their cells to tolerate low oxygen levels without triggering the harmful chronic polycythemia that other high-altitude populations develop. That EPAS1 variant came from the Denisovans. It is found in high frequencies in Tibetans and in much lower or absent frequencies in other populations. The Denisovans, who lived in Asia for hundreds of thousands of years, appear to have been present at high altitudes and to have developed this oxygen-sensing adaptation. When modern humans interbred with them, some of the descendants inherited the EPAS1 variant. Those descendants who moved to high altitudes in Tibet found that the Denisovan variant was extraordinarily useful. It spread rapidly through the Tibetan population by strong positive selection. This is one of the clearest examples of adaptive introgression in the human genome. A variant that evolved in one species under specific selective pressures in a specific environment was transferred to another species through interbreeding and then spread rapidly because it happened to be exactly what the recipient population needed. We're not just a product of our own genetic errors, we're a product of theirs too. Of the Neanderthals and the Denisovans and whatever other populations we interacted with across the long migration out of Africa and across the entire inhabited world. We borrowed their immune systems, their altitude adaptations, possibly their cold climate physiological variants. We took what we needed, not deliberately, not by choice, but by the random process of genes that happened to be useful spreading faster than genes that did not. We are more ancient than our species boundaries suggest, more connected to the other hominins than the term modern, human implies. The errors in our genome include errors that were not originally ours. The accidents that built us include accidents that happened in bodies that were not our ancestors, but that became, through interbreeding, part of our genetic heritage anyway. Let's come back to something personal, something intimate, something that the genetics ultimately points toward, but that is, in the end, about you specifically, and not about genomes in the abstract. You carry, in every cell of your body, approximately 3 billion base pairs of genetic sequence. Within that sequence are the errors we have been talking about. The broken GULO, the sRGAP2C duplication, the ARHGAP11B copy, the NOTCH2NL genes, the chromosome 2 fusion, the HAR sequences, the FOXP2 variant, the Neanderthal fragments, the Denisovan EPAS1 variant if you're of Asian or indigenous ancestry. All of the above plus thousands of variants that we have not discussed. Variants that affect your specific physiological traits, your specific disease susceptibilities, your specific cognitive tendencies, your specific emotional architecture. No other human being alive has exactly this combination. No human being who's ever lived had exactly this combination. You are, in a literal and specific genetic sense, unprecedented. The particular arrangement of errors and adaptations and inherited sequences that constitutes your genome has never existed before and will never exist again. And it is producing, right now, the brain that is processing these words. The experience of being you, right now, in this specific moment, is a unique and non-repeatable event in the history of life on Earth. Not in a spiritual sense, though it may be that too. In a strictly factual biological sense. There's never been another nervous system running on exactly these genetic instructions, in exactly this developmental history, with exactly this lived experience. The combination is singular. This is not an argument for narcissism. It is an argument against a certain kind of self-minimization. The tendency to feel that what is happening inside one's own experience is ordinary, unremarkable, a generic instance of a common category. The category is common. Human consciousness is common in the sense that there are billions of instances of it. But each instance is running on a unique genetic substrate, shaped by a unique developmental history, producing a unique configuration of experience that cannot be replicated anywhere else. The errors that built you are yours, not in the sense that you chose them or earned them, in the sense that they are the specific set of genetic accidents that constitute the physical basis of your specific consciousness. The brain that wonders about all of this is the product of every error in your particular genome, expressed in the particular developmental conditions of your particular life, producing the particular neural architecture that is right now, holding these thoughts. That is not nothing. It is in fact the only thing that actually exists in the first place. Not the genome in the abstract, not the species in the aggregate, just this. Just the specific experience of being you, right now in this moment, with all the errors and all the adaptations and all the borrowed sequences and all the consequences of all the accidents that built you over millions of years. There is a moment in human evolutionary history where everything nearly stopped, where the chain of compounding errors almost reached its end before it had gone anywhere significant. It happened approximately 74,000 years ago. A supervolcano on the island of Sumatra in what is now Indonesia erupted in an event now known as the Toba eruption. It was, by a substantial margin, the largest volcanic eruption in the last 2 million years. It ejected approximately 2,800 cubic kilometers of magma. It blanketed Southeast Asia, South Asia, and parts of East Africa in a thick layer of ash. It injected so much sulfur dioxide into the stratosphere that global temperatures dropped by as much as 15 degrees Celsius for years afterward. It was a volcanic winter that lasted potentially decades. Humanity nearly did not survive it. Genetic studies of modern human populations have identified what is called a bottleneck in our recent evolutionary history. A point where the effective population size of all modern humans dropped to an extraordinarily low number. Some estimates suggest as few as 10,000 reproductive adults. Some suggest as few as 3,000. These are the numbers you get when you look at the level of genetic diversity in modern human populations and compare it to what you would expect from a species of our size and age. We are genetically one of the least diverse large mammals on the planet, less diverse than chimpanzees, despite being far more numerous. Something reduced our ancestors to a tiny remnant population. The Toba eruption is the leading candidate for what that something was, though the debate about the exact size and cause of the bottleneck continues. What we know is that whatever happened, the entire genetic diversity of every human alive today, traces back to a group of survivors who made it through a period of extraordinary environmental stress. And here is what matters in the context of the errors. Every genetic variant in every human alive today is a descendant of the variants that were present in that tiny surviving population. The MYH16 deletion that made it through, the SRGAP2C duplicate that made it through, the NOTCH2NL copies that made it through, the FOXP2 variant that made it through, the chromosome 2 fusion, already fixed by then, that made it through. Whatever combination of accumulated errors existed in those few thousand individuals, that is what we all carry. The bottleneck did not create the errors. They had already been accumulating for millions of years before Toba. But the bottleneck almost erased them, almost eliminated the entire line that had been building them. And the fact that the line survived, by whatever margin of luck and adaptation and geography and resource access and social cohesion allowed those few thousand people to make it, is why you exist. Every human alive is the descendant of the people who happen to be in the right place, with the right combination of errors to survive the worst environmental catastrophe to hit the human species in the last 2 million years. That knowledge should produce something in you, not just intellectual recognition of a fact, something personal, because you are not just abstractly descended from those survivors. You are specifically the person you are, with the specific combination of inherited traits you have, because of who and where and how they were. Their survival was your precondition, not a distant, abstract precondition, the kind you can acknowledge and move past, a direct, specific, intimate one. The chain that runs from those survivors to you has been broken not once. In 74,000 years of unbroken biological continuity, every generation has found a way to persist, to reproduce, to pass forward whatever combination of errors and adaptations it was carrying. Through Toba and through every subsequent hardship, the line continued. That is the story inside your cells. Long before you existed as an individual, you were the purpose, in a purely functional sense, of an unbroken sequence of survival events going back millions of years. Here is something that tends to get left out of the cognitive evolution story. Something physical, something that connects the brain to the body in a way that matters for understanding what the accumulated errors actually built. Humans can run, not fast. No human is going to outrun a cheetah or a lion or even a dog over short distances, but humans can run for extraordinary distances at sustained speeds. The combination of our long stride, our bipedal gait, our exceptional thermoregulation through sweating, and our ability to breathe independently of our stride means that a fit human can sustain running speeds that would eventually exhaust quadrupedal prey animals over long enough distances. The evolutionary hypothesis is called persistence hunting. It has been documented in several surviving hunter-gatherer communities and appears to have been a significant food procurement strategy for much of human evolutionary history. A group of humans tracks prey across hot terrain, driving it at a sustained trot, following its tracks, pushing it to run repeatedly until the animal's thermoregulatory system fails. Quadruped animals pant to cool down, and panting requires them to breathe in synchrony with their stride, which limits how fast heat can be dissipated during a hard run. Human sweat from 2 to 4 million eccrine sweat glands distributed across essentially the entire body surface, a cooling system that operates independently of locomotion. Over long distances in hot conditions, humans can literally run prey animals to death by overheating. This capacity required a specific set of physical adaptations. The long Achilles tendon that stores and returns elastic energy with each stride. The gluteus maximus, the largest muscle in the human body, which stabilizes the trunk during bipedal running in a way that requires nothing comparable in other primates. The nuchal ligament, absent in chimpanzees, that stabilizes the head during running. The relatively hairless skin with extraordinary density of sweat glands. The narrow waist and broad shoulders that allow the upper body to counter-rotate with respect to the lower body during running, maintaining balance. And crucially, the brain that could plan a persistence hunt. That could track a specific animal over hours and kilometers. That could coordinate a group of hunters, communicating about the animal's direction, its condition, its likely path. That could maintain motivation and purpose through the sustained effort of a long chase. That could perform the probabilistic reasoning needed to predict where the animal would go. The physical adaptations for running and the cognitive adaptations for planning, coordinating and communicating were all part of the same evolutionary package. They co-evolved. A body built for persistence hunting required a brain capable of orchestrating it. A brain capable of orchestrating it required a body that could execute the plan over long distances. This matters for the genome story because it reminds us that the errors did not build a brain in isolation. They built a brain inside a body that was also undergoing changes, also adapting, also reorganizing itself in ways that made the brain's capabilities meaningful. The jaw that weakened and the skull that expanded and the brain that grew were part of a body that was simultaneously developing longer legs relative to arm length, more upright posture, better thermoregulation, hands that were freed from locomotion and available for tool use. The errors that built human intelligence were embedded in a physical transformation that was happening at the same time. The brain and the body were not evolving independently. They were part of the same selective process, driven by the same environmental pressures, producing changes that each made the other more viable. A bigger brain is only useful if the body can support it, feed it, and deploy its capabilities in ways that matter for survival. And the human body, as it was being built by selection, was creating exactly the platform on which the enlarged brain could be most useful. There is a specific aspect of human language that deserves more attention. Not just that we can produce speech, which is what FOXP2 contributed to, but what language does once it exists, what it makes possible that was not possible before. Language allows the transmission of information about things that are not present. This sounds simple, but consider what it means. Every other form of communication in the animal kingdom is fundamentally about what is present. A vervet monkey alarm call communicates information about a predator that is currently in the environment. A chimpanzee's submissive gesture communicates something about the current social relationship between two individuals. The signal and its referent are both present in the here and now. Human language can refer to things that are absent, to events that happened in the past, to states of affairs that might occur in the future, to possibilities that have never existed and might never exist. This displacement property of language, its ability to refer across time and space and into purely hypothetical domains, is what makes cultural accumulation possible. Before language with full displacement, knowledge died with the individual who held it. A chimpanzee who learned, through personal experience, that a certain plant cured a particular ailment had no way to pass that specific knowledge to the next generation in a durable form. The knowledge could not be encoded in anything that would survive the individual. It might be transmitted imperfectly through observed behavior, but the crucial causal structure, the understanding that this plant cures this ailment for this reason, could not be communicated. Language with displacement changes this entirely. A human who discovers something about plants, about the behavior of animals, about the layout of a territory, about the cause and effect relationships in their environment, can encode that knowledge in language and transmit it to other people, including people who were not present when the knowledge was acquired, including people who have not yet been born. This is what writing eventually made permanent. But oral traditions, before writing, were already doing something important. They were carrying knowledge across generations in a form that could be actively used by minds that never experienced the original events. The old woman in the group who had seen a drought 40 years ago and knew where the water sources persisted longest, who had described the signs that the drought was coming and the strategies that had worked, was a library that the group was accessing. Her language allowed her experience to shape the behavior of people who had not had her experience. Cultural memory extended the individual lifetime. This is the gift of displacement, and it required the full suite of cognitive and linguistic changes that the errors built. The working memory to hold complex propositional structures, the theory of mind to understand that you are conveying information to someone who does not have it, the executive function to construct coherent goal-directed narratives, the phonological precision that FOXP2 refined, the abstract symbolic capacity that is still not fully explained by any single genetic change, but that emerges from the integration of all of them. Human language in its full form is not just a more complex version of animal communication. It is a different kind of thing entirely. It is a system for encoding and transmitting arbitrary symbolic representations of anything, including things that do not and cannot exist at any time to any recipient in any context. And the cognitive architecture required to use it is the architecture that the errors built. That architecture took 3 million years to assemble from dozens of mistakes, and in the 50,000 years since it reached approximately its current form, it has transformed the planet. Now let's bring the threads together. Let's look at what all of these stories, told individually, add up to when you hold them all at once. There is a version of the human genome story that sounds like a just so story. Each mutation happened to be exactly what was needed at exactly the right time. The jaw weakened just as the diet was changing. The synaptic development slowed just as social structures were becoming supportive enough to sustain a long childhood. The brain expanded just as food sources were becoming calorie-rich enough to support the metabolic demand. Everything lined up, as if guided. But that is not what the evidence shows. The evidence shows a messier picture. A picture of multiple changes occurring in a context that was itself changing, each change sometimes working with the others, sometimes creating problems that had to be worked around, sometimes opening doors that the population was not immediately ready to walk through. The obstetric dilemma is not a happy story. It is a story of a brain that expanded faster than the birth canal could accommodate, and the result was, for all of evolutionary history before modern medicine, devastating mortality in childbirth. That is not a clever adaptation, that is a compromise. A situation where the benefits of brain expansion were large enough to be selected for despite the costs it imposed on reproductive physiology. The OTCH2NL vulnerabilities, The connection between brain expansion genes and autism and schizophrenia is not a happy story. It is the story of a genome pushed toward the edge of a functional range, where the margin between optimal and pathological has become dangerously narrow. That is not elegant design. That is the consequence of optimization for a single variable, brain size, at the expense of robustness. The Neanderthal-derived depression and anxiety risk variants are not a happy story. They are the story of a species carrying, for reasons of historical contingency and ancient interbreeding, adaptations that fit an environment that no longer fully exists. That is not purposeful construction. That is the accumulation of baggage that could not be discarded fast enough. And yet, the brain that emerged from these compromises and vulnerabilities is extraordinary, not despite the compromises, not despite the vulnerabilities. Because of them, the same gene variants that create the risk of autism in altered copy number are the ones that expanded the cortex. The same extended critical period that makes human childhood so long and so dependent is the one that allows the depth of learning and cultural transmission that makes human civilization possible. The same displacement property of language that allows the transmission of abstract knowledge also allows the elaboration of elaborate fictions that have no basis in reality, some of which cause serious harm. The human genome is not a success story in the sense of a plan well executed. It is a success story in the sense of a process that kept going, that kept finding paths through the space of possible genomes, that kept encountering environments in which the accumulated errors added up to something that could survive and reproduce. And that process produced, as a byproduct, not just survival and reproduction, but consciousness, culture, and the capacity for the kind of understanding that includes understanding itself. That is not what evolution is supposed to do. Evolution is supposed to produce organisms that survive and reproduce. It is not supposed to produce organisms that ask why they survive and reproduce. It is not supposed to produce organisms that develop the concept of evolution and use it to reconstruct their own history. It is not supposed to produce organisms that write music or prove theorems or grieve for strangers. And yet here we are, the accidental complexity, the unintended depth, the product of errors that kept compounding in a direction that, as far as the universe was concerned, had no meaning or destination, but that, from the inside, feels like a great deal of both. There is a protein domain in your genome called DUF1220. The name tells you almost nothing useful, which is often how genomic discoveries get named when the people who find them do not yet understand what they are looking at. DUF stands for Domain of Unknown Function. It was named before its function was understood. By the time researchers did understand its function, the name had stuck. DUF1220 protein domains are encoded by a family of genes called the NBPF genes. These genes have undergone dramatic copy number expansion in the human lineage. The number of copies of DUF1220 domain encoding sequences varies across the primate family, and the variation follows an extraordinarily clean pattern. Macaques have approximately 32 copies of DUF1220 encoding sequences. Chimpanzees have approximately 49 copies. Gorillas have approximately 66 copies. Humans have approximately 272 copies. 272 in a diploid human genome. The correlation with brain size and cognitive complexity across the primate phylogeny is striking. More copies of DUF1220 corresponds with larger brains and more complex cognition, This does not mean that copy number is the only determinant, or even the primary determinant, but the correlation is strong enough and consistent enough that it cannot be dismissed as coincidence. Researchers discovered that DUF1220 protein domains are specifically expressed in neurons of the human neocortex. The regions with the highest expression are specifically the neurons in layers 3 and 4 of the cortex, the layers most associated with complex associative processing. And the expression pattern shows something important. DUF1220 appears to be involved in the proliferative behavior of neurons, in driving increased dendritic development, in the expansion of neuronal processes that forms the basis of synaptic connectivity. When the copy number is perturbed in humans, the results follow the same pattern we have seen with NOTCH2NL. Too many copies of certain DUF1220 encoding regions is associated with macrocephaly and autism. Too few copies is associated with microcephaly and schizophrenia. The same functional tension, the same narrow range where things work, flanked on both sides by pathology. What the DUF1220 story adds to the picture is scale. The human genome did not just acquire one or two key brain expansion features. It acquired them redundantly, at multiple levels, through multiple independent mechanisms. The MYH16 deletion, the sRGAP2C duplication, the ARHGAP11B expansion, the NOTCH2NL duplications, the DUF1220 expansions, the human accelerated regions, the FOXP2 refinement. All of them pointing in the same direction. All of them contributing to the same outcome. Not because they were designed to work together, because the selection pressure that allowed each one to persist and spread was the same selection pressure, increasing brain complexity, increasing cognitive capacity, increasing behavioral flexibility. This convergence is what makes the human genome story so extraordinary. Not that one mutation changed everything, but that dozens of mutations, many of them independently and accidentally, all happened to push in a direction that the environment was rewarding. A direction that was, in the environments of 3 million years of hominin evolution, increasingly advantageous. And so each one spread, and each one found itself in a genome that already carried the others, and together they compounded in ways that none of them could have produced alone. Let's talk about starch. This might seem like a strange detour from the story of brain evolution, but it is not a detour. It is another piece of the same story, and it connects in ways that matter. The human genome contains a gene called AMY1. AMY1 encodes salivary amylase, the enzyme that begins the digestion of starch in the mouth. You have experienced this if you have ever held a piece of bread in your mouth for a few seconds and noticed that it starts to taste slightly sweet. That is salivary amylase beginning to break starch down into sugars before you even swallow. Most primates have a few copies of AMY1. Chimpanzees typically have 2 copies per diploid genome. Most non-human primates have somewhere between 2 and 4. Humans typically have somewhere between 6 and 8 copies, though the number is variable, and some individuals carry as many as 15 copies. More copies of AMY1 means more salivary amylase. More salivary amylase means more efficient starch digestion. More efficient starch digestion means that cooked and processed starchy foods, tubers, grains, legumes become a significantly more calorie-accessible food source. The increase in AMY1 copy number in the human lineage is estimated to have occurred roughly 1 million years ago, with some evidence for the expansion predating and accompanying the use of fire for cooking. Cooked starchy foods are extraordinarily calorie-dense and calorie-accessible. A raw sweet potato provides only about a third of the calories that the same sweet potato provides when cooked, because cooking breaks down the cell walls and gelatinizes the starch, making it vastly more available to the digestive enzymes. The human brain is expensive. It consumes approximately 20% of the body's resting metabolic rate, while accounting for only about 2% of body weight. That disproportionate energy demand means that feeding the brain is a serious nutritional challenge. In environments where calorie availability is uncertain, a brain this large is a metabolic liability as much as it is a cognitive asset. You have to eat enough to keep it running. The AMY1 expansion helps solve that problem. By increasing the efficiency with which humans could extract calories from cooked starchy foods, it helped make the brain economically viable. The dietary shift toward cooked, processed, calorie-dense starchy foods was not just a consequence of brain evolution and tool use. It was one of the enabling conditions of it. A feedback loop in which bigger brains enabled better food processing, and better food processing provided the calories to sustain bigger brains. And this is another error, in the sense of an accidental genomic duplication that pushed in the same direction as all the others. The AMY1 copy number expansion was not random in the sense of having no fitness consequence. It was selected for. Having more copies of AMY1 was better in the dietary environment where cooked starchy foods were available. But the initial duplications were accidents, random errors in DNA replication that happened to produce extra copies of a gene whose extra copies happened to be useful. The brain that was running on those extra calories, that was being maintained by the starch processing efficiency that AMY1 expansion provided, was the same brain that was being expanded by ARHGAP, 11B, and NOTCH, 2NL, and SRGAP2C. The errors were all co-occurring, codepending, co-enabling. The dietary accident and the neural accidents and the regulatory accidents all made each other possible. Here is something to think about, something that is easy to miss when you focus on the individual molecular stories. Every single one of these mutations appeared initially in one person, one individual, one brain, one set of cells that happened to carry a new error that no member of their species had carried before. That individual did not know they were different. They could not sequence their own genome. They could not know that the error would persist and spread and compound with other errors and build towards something unprecedented. They just lived. They reproduced. They passed something forward. And that individual was also, almost certainly, unexceptional in every other way. Not a genius, not a visionary, not a founder in any heroic sense. Just an individual in a population, carrying an error, managing to survive and reproduce. The significance of that error was entirely invisible from the inside, entirely invisible from any perspective available at the time. This is what evolutionary change looks like from the inside of the history it is making. Invisible. A completely ordinary sequence of events, nothing obviously remarkable about any of it, that turns out to have been the exact sequence that changed everything. How many times has this happened? How many evolutionary turning points have looked, from the inside, like nothing at all? How many individual animals, in how many lineages, have carried mutations that were the first instances of changes that became defining features of their descendants, without any signal, any awareness, any sense that this was a different moment from all the others? The answer is all of them. Every evolutionary change was first an ordinary day in the life of an ordinary individual who happened to carry a new version of something. Every species was preceded by an individual who carried the first mutation in that direction, who lived and died without knowing what they had started. And this continues. Right now, somewhere in the 700 million individuals born in this year, there is a person carrying a mutation that did not exist before. A mutation that might turn out to be nothing, or a mutation that, in combination with mutations that will appear in future generations, becomes the foundation of something that the human lineage has not yet been. Something that people in 1,000 years will look back and trace to this moment, to this ordinary person living an ordinary life, carrying an error they do not know about. Evolution does not announce itself, it just keeps going. Now let's talk about the social brain, about the specific cognitive architecture that the errors built that most distinguishes human psychology from the psychology of other animals. Other primates are social. Chimpanzees live in complex social groups with intricate dominance hierarchies, coalition politics, and individual relationship histories that persist over years. They remember who did what to whom, and they adjust their behavior accordingly. They have politics in a real sense. They form alliances, they manipulate social information, they use deception. Social cognition in chimpanzees is sophisticated and impressive. But human social cognition runs at a different level of complexity, not just in degree, but in kind. And the difference is captured in a concept that developmental psychologists call theory of mind. Theory of mind is the ability to attribute mental states to other individuals, to understand that other people have beliefs, desires, intentions, and knowledge that are different from your own, and to reason about what other people are thinking and why they are doing what they are doing, based on a model of their mental states, not just their behavior, their mental states. Humans develop theory of mind around age 4. Before 4, children consistently fail tasks that require them to reason about other people's beliefs, specifically other people's false beliefs, beliefs that the child knows to be incorrect. The Sally Ann task is the classic example. A child watches Sally hide a marble in a basket, then leave the room. While Sally is gone, Anne moves the marble to a box. Sally returns. Where will Sally look for the marble? Children under 4 say in the box where the marble actually is, because they cannot separate their own knowledge from Sally's knowledge. After 4, they say in the basket where Sally thinks the marble is, because they understand that Sally has a false belief, and that her behaviour will be driven by her belief rather than by the objective location of the marble. Chimpanzees show limited versions of theory of mind in some experimental paradigms, but fail consistently in others. They can track what another individual has seen or not seen, and adjust their behaviour accordingly. But they do not fully reason about other agents' beliefs in the way that human 4-year-olds do. Full theory of mind is the foundation of everything that makes human culture complex. Language is only fully effective when you understand what your interlocutor knows and doesn't know, so you can calibrate what information needs to be made explicit. Teaching is only effective when you understand what the learner currently understands, so you know where to start. Deception requires modelling what the target of the deception currently believes, and crafting a narrative that will mislead that specific belief. Cooperation at scale among individuals who are not close kin requires the ability to model other people's intentions and commitments, and to reason about whether they can be trusted. All of this runs on theory of mind, and theory of mind runs on brain structures, particularly in the prefrontal cortex and in the temporoparietal junction, that are significantly more elaborate in humans than in other primates. The same brain structures that the gene expansion events of the last 3 million years built. There is a specific brain region called the default mode network. It is a set of interconnected regions in the prefrontal, parietal, and temporal cortex that is active when the brain is not focused on an external task. When you are resting, daydreaming, thinking about the past or the future, imagining other people's perspectives. For a long time, neuroscientists thought this network was doing nothing useful. It was the brain idling. But it is actually doing something extraordinarily sophisticated and specifically human. It is the network of self-referential processing, of autobiographical memory, of social reasoning, of mental time travel, of perspective taking. The default mode network is proportionally larger in humans than in other primates. It develops slowly, becoming fully interconnected only in the mid-20s, exactly tracking the extended critical period that sRGAP2C and the related developmental changes created. And it is the neural substrate for all the capacities that feel most distinctively human. The ability to think about yourself, thinking. The ability to imagine other times and other perspectives. The ability to construct a narrative identity, a sense of self that extends across past and future, rather than being confined to the immediate present. Every time you worry about something that has not happened yet, you're using the default mode network to simulate a possible future. Every time you remember a past event and reflect on it, you are using the default mode network to reconstruct the past. Every time you try to understand why someone did what they did, or imagine how they feel, you're using the default mode network to model another mind. Every time you feel the specific kind of loneliness that comes from feeling unseen or misunderstood, you're experiencing the default mode network's model of the discrepancy between how you think you are being perceived and how you want to be perceived. This network, this most human of neural structures, was built by the errors, by the duplications and regulatory changes that expanded the cortex, by the developmental slowdown that extended the critical period during which its complex connectivity is established, by the combination of accidents that push the hominin brain in the direction of greater social complexity, greater self-modeling capacity, greater capacity for perspective taking and mental time travel. The default mode network is also the network that goes wrong in depression. When the simulation of the future becomes dominated by negative predictions, when the reconstruction of the past becomes dominated by rumination, when the model of other people's minds becomes dominated by the fear of rejection or judgment, you have the neural substrate of human depression and anxiety, the same network that enables the highest forms of human social intelligence and self-reflection, also enables the specific forms of suffering that are uniquely human. You cannot take the default mode network and subtract the capacity for depression while keeping the capacity for meaning-making and social understanding. They are the same network, operating in different modes. The vulnerability and the capacity are neurologically inseparable. Think about what it would have been like somewhere in the Middle East, roughly 70,000 years ago. Two groups meeting at a water source. One group has come from Africa, carrying the accumulated genetic legacy of the brain expansion genes and the FOXP2 language mutation and the HARs and all the rest. The other group has been in the region for hundreds of thousands of years, carrying their own adaptations, their own immune toolkit, their own evolutionary history. They look at each other. They are both human in any meaningful sense. They both have language. They both use fire. They both bury their dead. They both make art. Whatever happened next is not in the written record because there was no writing. But the genetic record tells us some of it. Some of them became part of the same community. Some of their children survived. And those children's children's children are some of us. The human genome is a record of who we encountered and what we inherited from them, not just who we were. The language gene is called FOXP2. That name is dry and clinical and does absolutely no justice to what it does or what it means. FOXP2 is a transcription factor, which means it is a protein that binds to DNA and regulates the expression of other genes. Specifically, it binds to the promoter regions of a large number of genes involved in the development and function of brain regions associated with movement, planning, and language. It is expressed during fetal development in regions of the brain that will become critical for speech production. The gene itself is ancient. It is present in fish, birds, crocodiles, mice, chimpanzees. Mutations in FOXP2 in mice cause abnormal ultrasonic vocalizations in pups. Mutations in FOXP2 in songbirds severely disrupt their ability to learn the complex vocalizations that are essential for their behavior. In humans, a specific mutation in one copy of FOXP2 causes a syndrome that includes severe difficulty with the precise motor control required for speech, and with various aspects of grammar and language comprehension. The affected family members, studied extensively by Anthony Monaco and colleagues at Oxford in the 1990s, had a mutation in exactly one copy of FOXP2 and showed consistent specific language impairments across 3 generations. The human version of FOXP2 differs from the chimpanzee version in exactly 2 amino acids. 2 out of 715. Those 2 changes are specific to the human and Neanderthal lineages. They were not present in the ancestral hominid population. They evolved in the lineage after the human and chimpanzee lineages diverged, and they appear to have been positively selected, spreading rapidly through the human population because they were advantageous. When researchers inserted the human version of FOXP2 into mice, replacing the mouse version with the 2 amino acid differences from the human sequence, the mice showed changes in brain circuitry in the striatum and cerebellum. They showed changes in their ultrasonic vocalizations. They showed better performance on certain learning tasks that depend on the basal ganglia circuitry that FOXP2 affects. The changes were not dramatic. Mice with human FOXP2 did not suddenly start talking, but the changes in the circuitry were real and measurable. What the human version of FOXP2 appears to have done is to refine and extend the function of brain circuits involved in the learning and execution of complex, precise, sequential motor programs. Speech is a sequential motor program. The specific, precise, rapid coordination of lips, tongue, palate, larynx, and diaphragm that is required for speech is one of the most demanding motor tasks the human nervous system performs. The human FOXP2 appears to have made the neural circuits for this kind of coordinated sequential movement more sophisticated, more capable, more plastic. But FOXP2 does not encode language. It does not encode grammar or vocabulary or the ability to use symbols to represent absent things. Those capacities involve many genes, many circuits, many processes that FOXP2 is only part of. What FOXP2 appears to have contributed is something more specific, a refinement of the motor circuitry that makes the physical production of speech possible. the neural foundations for the precise, rapid, learned motor control that allows you to produce dozens of distinct phonemes per second, to modify their acoustic properties in ways that carry grammatical information, to coordinate the complex dance of vocal anatomy that makes spoken language physically possible. Without that motor foundation, language as we know it, spoken language, the kind that can be produced rapidly and with enough acoustic precision to convey complex information, would not work. You could have the conceptual apparatus for language, the symbols, the grammar, the working memory, the social motivation to communicate, but if you could not physically produce the sounds with sufficient precision, you would not use it in the way that human language is actually used. FOXP2 helped build the body of language. 2 amino acid differences from the ancestral version. 2-point mutations in a 715 amino acid protein that had been under purifying selection for hundreds of millions of years. 2 errors that, in the context of the circuits they regulated, and the selection pressure of a species increasingly dependent on communication, turned out to be the molecular foundation of the human voice. Think about that the next time you speak. Every word you produce depends on motor circuits that would not exist in their current form without 2 specific errors in a transcription factor gene. Now let's talk about something that most people do not connect to brain evolution, but that is directly related: cancer. Humans get cancer at rates that are, in some respects, unusual compared to our close relatives. Not all cancers, not uniformly, but there are specific categories of cancer, specific tumor types, that are more common in humans than in other great apes, and some of them appear to be connected directly or indirectly to the same genetic events that expanded the human brain. The connection starts with a class of genes called proto-oncogenes. These are normal cellular genes that, when they are mutated or inappropriately activated, can drive uncontrolled cell proliferation. They are essentially accelerators for cell division. In normal cells, they are tightly regulated. They turn on when cells need to divide and turn off when division needs to stop. When that regulation fails, when a proto-oncogene gets stuck in the on position, cancer can follow. The Notch genes are proto-oncogenes. The Notch signaling pathway, the same pathway that the Notch2NL duplications influence to expand the cortex, is also involved in driving cell proliferation in many tissue types throughout the body. Notch pathway mutations are found in a significant fraction of human cancers, including leukemias, breast cancers, and several others. The same molecular machinery that, in the developing brain, allows neural progenitor cells to remain in a proliferative state for longer, producing more neurons before differentiating, can, in a different context, become the driver of malignant proliferation. This is not a coincidence. It is a fundamental trade-off. Evolution could not give us a gene that drives neural progenitor cell proliferation during brain development without giving us a gene that, under the wrong circumstances or with the wrong mutations, drives abnormal cell proliferation elsewhere. The story of elephants makes this even clearer. Elephants almost never get cancer. Their cancer rate, despite their enormous body size and decades-long lifespans, is dramatically lower than the cancer rate in humans. Why? Because elephants have 20 copies of the TP53 gene. Humans have 1. TP53 encodes the protein p53, which is one of the most important tumor suppressors in the genome. p53 monitors DNA damage in cells. When it detects damage, it can trigger cell cycle arrest, forcing the cell to either repair the damage before dividing or to undergo apoptosis, programmed cell death rather than proliferating with damaged DNA. Having 20 copies of TP53 means that elephant cells are extraordinarily sensitive to DNA damage. Any cell that accumulates mutations is very likely to be detected and eliminated before it can become a cancer. Having one copy, as humans do, means that a single mutation to that one copy can be enough to disable the surveillance system in a cell, allowing subsequent mutations to accumulate without triggering cell death. Why do humans have only one copy? Why did evolution not give us more TP53, more cancer protection, especially given how long-lived we are and how much metabolic activity our enormous brains require? The honest answer is that we do not fully know. But one possibility is that having more TP53 creates its own trade-offs. P53 activation during development can trigger apoptosis in progenitor cells, and if the system is too sensitive, it might eliminate cells that should be dividing to build the brain. The same cells that notch 2NL and ARHG-AP11B are telling to keep dividing to produce more neurons. There may be a fundamental tension between the cellular surveillance that prevents cancer and the proliferative drive that built the large human brain. More P53 might mean fewer cancers and also fewer neurons. If this is correct, if there is a real trade-off between brain expansion and cancer resistance, then human vulnerability to cancer is not a mistake. It is an indirect consequence of a choice, if you can call an evolutionary process a choice, to maximize brain size at the cost of reduced protection against cellular proliferation errors. We are more prone to cancer than elephants, partly because our brains drove our cells toward proliferation in ways that could not be perfectly insulated from malignant outcomes. The same cellular biology, the same molecular machinery, the cancer vulnerability and the brain expansion are not separate stories. They are the same story with two different endings. Now let's look at one final piece of evidence that our genome was built by accident. Evidence that is, if you think about it, visible in every cell in your body, in the fundamental structure of the chromosomes themselves. Humans have 46 chromosomes. Chimpanzees, gorillas and orangutans have 48. This is not a typo and it is not a mistake. Non-human great apes have 2 more chromosomes than humans. They have 48. We have 46. For a long time, this was a puzzle. You cannot simply lose a chromosome and survive. Chromosomes carry genes. If you lose a chromosome, you lose all the genes on it, and that would be catastrophic. So how did the ancestor of modern humans go from 48 chromosomes to 46? Where did the 2 chromosomes go? The answer is that they did not go anywhere. They fused. Human chromosome 2, the second largest chromosome in the human genome, is a direct fusion of 2 ancestral chromosomes that correspond to chimpanzee chromosomes 2A and 2B. They are still there, all of their genes still present, still functional. They just got joined end to end into a single chromosome instead of existing as 2. We know this because of the signature the fusion left in the DNA sequence. When chromosomes get fused, the joining leaves a specific molecular scar. Normally, the ends of chromosomes, the telomeres, are capped with specific repetitive sequences. But when 2 chromosomes get fused end to end, the telomeric sequences from both ends get buried inside the resulting chromosome. They're no longer at the end. They are in the middle. And that is exactly what you find when you look at human chromosome 2. In the middle of the chromosome, there is a region of degenerate telomeric sequence, the remnants of the old chromosome caps from the fusion event. Proof of the joining. There is also a vestigial centromere. Each normal chromosome has one centromere, the constriction point that the spindle fibers attach to during cell division. The fused chromosome 2 still has one functional centromere, the one from the ancestral chromosome 2b. But there is also a remnant of the other centromere from ancestral chromosome 2a, now inactivated, sitting in the middle of chromosome 2 as molecular evidence of what it used to be. The fusion event happened somewhere between 3 and 6 million years ago, in the hominin lineage after it separated from the chimpanzee lineage. We do not know exactly when or exactly why. The most likely explanation is that the fusion was initially a neutral or mildly disadvantageous event in a small population. An individual was born with the fusion, probably had slightly different fertility because chromosomal rearrangements can cause problems in meiosis, but the individual survived and reproduced. The population was small enough that genetic drift allowed the fusion to spread. Eventually it became fixed. The fusion itself is not known to cause any direct change in gene function. All the genes are there. They still work. What changed is the chromosome number, and that matters because when two individuals who are heterozygous for the fusion, meaning one has the fused version and one has the original two separate chromosomes, try to have offspring, the meiosis gets complicated. It can produce offspring with the wrong number of chromosomes. This means the fusion event, when it first appeared, would have created a reproductive barrier between individuals who carried it and individuals who did not. A partial barrier, not an absolute one, but enough to complicate mating and to produce some offspring with chromosomal abnormalities. In a small enough population, the fusion could spread until it was fixed, meaning everyone carried it, and then the reproductive barrier would disappear because everyone would be on the same side of it. The chromosome 2 fusion is, in a sense, the most literal evidence that the human genome was not designed. A designed system does not fuse two chromosomes together and leave the molecular scar of the joining sitting in the middle of the genome for every geneticist to find 3 million years later. A designed system does not accidentally reduce the chromosome number of an entire species through a random chromosomal rearrangement that happened to spread through a small population. This is exactly what an accidental process looks like. The scar is right there. The evidence is in every cell of your body. And yet here we are. The species with 46 chromosomes instead of 48, carrying the molecular record of the accident that changed our chromosome number, building civilizations and composing symphonies and sequencing our own genomes and finding the evidence of our accidental origin written in the code that runs every cell we have. So what does all of this add up to? What is the synthesis? What does it mean that the human genome is built from accumulated errors, from broken genes and accidental duplications and chromosome fusions and regulatory rewiring and borrowed sequences from other species? It means something both humbling and extraordinary. Let's take each in turn. The humbling part is this. Humans are not the culmination of an intentional process. There is no evidence in the genome for design, for direction, for any kind of forward-thinking optimization. The errors that built us did not know they were building us. They were just accidents, random variations in a copying process that happened to encounter an environment and a selection regime that allowed them to persist and compound. The jaw muscle mutation did not know it was making room for a bigger brain. SRGAP2C did not know it was extending the critical period for learning. ARHGAP11. B did not know it was building the cortex that would someday try to understand what ARHGAP11B was. There is no plan in the human genome. There is only the accumulated record of what survived, what worked well enough, in the environments that existed, in the populations that existed, at the moments when each mutation appeared to propagate forward to the next generation. And what worked is, by any objective measure, astonishing. But it was not aimed at being astonishing. It aimed at nothing. It just survived. The extraordinary part is this: none of this was inevitable, and all of it was possible. This specific combination of errors that built the human brain required a specific sequence of events at specific moments. If the MYH16 mutation had appeared 1,000,000 years earlier, before the dietary shift and before the social structures that could absorb a weaker jaw, it probably would have been eliminated. If SRGAP2C had appeared in a different lineage, one that had not already developed the social structures to support a long childhood, it might not have been advantageous. If ARHGAP11B had appeared in a lineage where the skull was not already expanding to accommodate a larger brain, the effect might have been pathological rather than constructive. The errors that built human intelligence were possible, in the sense that the preconditions for them were met because of what had already happened, because of earlier errors and earlier adaptations that had already shifted the selection landscape in a particular direction. The human genome is not the result of a plan, but it is also not the result of pure random luck. It is the result of an accumulation of contingencies that, having each occurred, made the next step possible in a way it would not otherwise have been. That is what evolution looks like from the inside of the genome, from the inside of the species that the errors built. A cascade of accidents that was possible because of what came before, and that made what came after possible. Not inevitable, not designed, just possible, and then actual. And here is the thing about the specific errors we have been talking about. They have not stopped. The human genome is still accumulating mutations. We're still making errors in our DNA, copying every time a cell divides. The question of which errors are being selected for right now, which are being eliminated, which are being spread through the population by drift or by change selection pressures, is a question that will take thousands of years of future evolution to answer. We cannot see the present from inside it. What we can see is the pattern of the past. And the pattern of the past tells us that the most important things about us were accidents. Not cosmic accidents in the sense of being meaningless, but accidents in the literal sense of having happened without intention, without design, without any awareness on the part of the molecules involved of what they were building. The brain that is reading these words right now, or listening to them, is the product of that process, of those specific errors in that specific sequence, of the MYH16 deletion and the SRGAP2C duplication and the ARHGAP11B duplication and the Notch2NL expansions and the HAR rewiring and the FOXP2 amino acid changes and the chromosome 2 fusion and the Neanderthal immune sequences and the broken GULO gene and dozens of others we have not even discussed. All of them contributed, in ways that interact in ways we are still mapping, to the neural architecture inside which you are experiencing whatever it is you are experiencing right now. There is one more question, the deepest one, the one that all of the genetics ultimately points toward but cannot answer. What does it mean that the errors produce something that is aware of itself, not just intelligent, not just capable of complex behavior, aware, self-aware, capable of looking inward and asking what is happening in there capable of reading a description of its own genetic origins and feeling something about them. Curiosity maybe, or wonder, or a kind of vertigo at the recursiveness of it. You are a brain that was built by errors, understanding that it was built by errors, and feeling something about that understanding. That is not a simple thing. No other animal we know of has a brain that does that. Every other animal experiences its world, processes information, makes decisions, feels pain and hunger and social belonging. But the loop of self-referential awareness, the ability to hold your own existence as an object of contemplation, the ability to ask not just what is out there, but what am I, and what does it mean that I am asking that appears to be uniquely human? And the genetic story does not explain it. Not fully. We know which gene duplications expanded the cortex. We know which regulatory changes rewired its architecture. We know which mutations refined the circuits for language and sequential motor learning. But we do not know, at a mechanistic level, how any of this produces awareness. How the physical process of neural computation produces the experience of there being someone for whom the computation is happening. This is called the hard problem of consciousness, and it is genuinely unsolved. Not unsolved in the sense that we just need more data, unsolved in the sense that the conceptual framework for answering it may not yet exist. We can describe the neural correlates of consciousness, the patterns of brain activity that accompany conscious experience. We can build models of how information is integrated and broadcast in the brain in ways that seem relevant to conscious processing, but we cannot bridge the explanatory gap between the physical description and the experiential reality, between the neurons firing and the felt quality of firing. What we can say is that the errors that built the human brain built something that, at some point in evolutionary history, crossed a threshold below which there was processing and behavior without awareness, and above which there was processing and behavior with awareness. We do not know exactly where that threshold is, or what specifically crossed it, or whether it was crossed gradually or in a more punctuated way. What we know is that A, R, H, G, AP, 11B, and NO, TCH2NL, and SRG, AP2C, and FOXP2, and the HARs and all the rest built a cortex large enough, complex enough, integrated enough, and neurologically immature enough at birth to require decades of experience-dependent development, and that at the end of that process, there is something it is like to be you. There is experience. There is awareness. There is a felt sense of existing in a moment, of having a past and a possible future, of being a self that is distinct from the world while being embedded in it. That is what the errors ultimately built. Not just intelligence, not just language, not just the capacity for abstract thought, the capacity for the experience of having those things, for knowing that you know, for feeling that you feel, and we have no idea how. We have the genetic story of how the hardware was built. We do not have the story of how the hardware became capable of asking why it was built. Maybe that question will be answered someday. Maybe it will turn out to have a clean mechanistic explanation that we simply have not found yet. Maybe the hard problem of consciousness will dissolve once we understand the computational architecture of the brain in sufficient detail. Maybe the gap between the neural description and the experiential reality will close as the science gets better. Or maybe it will not close. Maybe there is something about the first-person perspective, about the subjective character of experience, that cannot be fully captured in third-person descriptions of neural processes, no matter how detailed. Maybe the fact that consciousness is intrinsically perspectival, intrinsically seen from the inside, means that an external description of it will always leave something out. If that is true, if consciousness is not fully reducible to its physical substrate, then the errors that built the human brain did something that transcends the genome entirely. They built the conditions in which something genuinely new, something that had not existed in the universe before, could emerge. Not just a more powerful information processing system, something that experiences being a more powerful information processing system, something for which there is a what it is like, and that is either the most important thing that has ever happened on this planet, or it is the most important thing we have not yet found a way to properly describe. Possibly both. Now let's think about what all of this means for how you move through the world, not in an abstract sense, in a practical personal sense. You carry NOTCH, 2NL duplications that expanded your cortex, and that also, in other dosages in other people, contribute to autism and schizophrenia. You carry SRGAP2C that slowed your synaptic development and extended your critical period and made you capable of the kind of learning you have done throughout your life. You carry Neanderthal-derived sequences in your immune system that may be keeping you alive, and possibly other Neanderthal-derived sequences that nudge your risk of depression or anxiety in one direction or another. You carry the broken GULO gene that means you depend on vitamin C from your diet. You carry FOXP2 variants that refine the neural circuits for speech. You carry ARGAP11B that expanded your cortex. You carry the molecular scar of the chromosome 2 fusion. You carry all of this, and you also carry the vulnerabilities that come with it. The human brain is extraordinary, and it is also fragile. It is extraordinarily demanding of metabolic resources. It takes about 20% of your body's total caloric intake to run. It is extraordinarily sensitive to developmental conditions, to the quality of early relationships and environments, to trauma and to deprivation. It is extraordinarily slow to mature, leaving you vulnerable and dependent for longer than any other animal. It is extraordinarily capable of suffering in proportion to its extraordinary capacity for experience. The mental health crisis that is visible everywhere you look in modern societies is not a sign of weakness in the people experiencing it. It is at least in part a sign of what the brain was built to do, and what happens when the conditions it was built to operate in are not present. Brains built for deep social connection, struggling in environments of isolation. Brains built for moderate episodic stress, struggling with chronic, pervasive background anxiety. Brains built for a world of meaningful challenges and clear threats, misapplying that threat detection system to social evaluation and economic uncertainty and futures that cannot be controlled. The architecture is ancient. The environment is new, and the mismatch creates suffering that is real, and that is not anyone's fault. The errors that built the human brain could not have anticipated modern environments because there is no anticipation in evolution. They built for the average selection pressures of millions of years of hominin evolution. They built a brain that was extraordinarily well adapted to a world of small groups, immediate threats, complex social relationships, and the kind of learning that happens through direct experience. And they gave that brain enormous plasticity, the capacity to adapt to new conditions through individual development rather than genetic change. That plasticity is what allowed humans to spread across the planet, to live in Arctic conditions and tropical rainforests and high-altitude deserts, to develop dozens of different subsistence strategies to build extraordinary cultural diversity across thousands of populations. But plasticity has limits, and when the environment changes faster than the plasticity can adapt, when it changes within a single generation rather than across many, there is friction, there is suffering. Understanding this does not make the suffering go away, but it changes how you hold it. Your vulnerability, and every human being's vulnerability, is not a flaw in an otherwise well-designed system. It is part of the same system that generates your capacity for joy, for meaning, for connection, for the kind of interior richness that no other animal we know of experiences. You cannot subtract the vulnerability and keep the richness. They are built from the same material. They are the same errors, seen from two different angles. There is one more thing I want to tell you about. It is something that does not get talked about enough when people discuss human evolution and human uniqueness. It is about what happens between generations, about how the errors that built the brain created the conditions for something that transcends genetics entirely. The human brain, extended beyond anything the genome could directly encode, created culture. Not culture in the weak sense of habits and preferences. Culture in the strong sense. Accumulated knowledge, refined over generations, each generation inheriting the intellectual work of every generation before it, adding to it, correcting it, passing it forward in an ever-growing archive of understanding. A chimpanzee can learn to crack nuts with a stone in a specific way that is passed on through imitation within a community. That is a form of cultural transmission. But it does not compound. The knowledge does not grow across generations. A chimpanzee born today does not have access to the accumulated nut-cracking wisdom of 1,000 generations of nut-cracking ancestors, refined and corrected and expanded over time. The information resets with each generation. It is maintained but not built upon. Human culture compounds. The reason you know that the Earth orbits the Sun is not that you personally worked it out. It is that a long chain of thinkers, stretching back through centuries, each one building on the work of the ones before, eventually worked it out collectively, and that collective work was recorded and transmitted, and is now accessible to you as part of your cultural inheritance. You stand on the shoulders not of one giant, but of an uncountable number of ordinary people, each of whom added something small, each of whom built on everything that came before. This compounding is only possible because of the brain that the errors built, because of the language capacity, the extended critical period, the theory of mind, the working memory, the capacity for abstract symbolic thought. And it is only possible because of the extended childhood, which creates a long enough window for cultural transmission to happen fully, for the next generation to receive and consolidate the accumulated knowledge of all previous generations before they need to start contributing to it themselves. The genome built the brain that built the culture, and then culture took over in a way that evolution by natural selection had never encountered before. Because culture is a form of inheritance that is much faster than genetic inheritance. A genetic mutation takes hundreds or thousands of generations to spread through a population. A cultural innovation can spread in a single generation, and once a cultural innovation is established, it can be refined and built upon in real time by living minds, rather than waiting for random mutations to improve on it. This is why human technological progress accelerated so dramatically once it reached a certain threshold. Not because human brains got better in recent millennia, they did not. The genome has not changed significantly in 50,000 years. What changed is that the accumulated cultural knowledge crossed a threshold where enough pieces existed to enable rapid synthesis and combination, where writing allowed knowledge to persist beyond individual memory and to be accessed by minds that were never in the same place at the same time, where the printing press amplified the reach of each recorded thought, where digital communication made the accumulated thoughts of every living mind simultaneously available to every other. The errors that built the human brain built the runway on which culture took off, and culture then flew at a speed that the errors could not have anticipated, in a direction that the genome had no instructions for, toward a complexity of human understanding that is still accelerating. You are both the product of those errors and the current furthest point in the process they started, the part of the universe that has most fully developed the capacity to examine what it is and where it came from, and what the accumulated accidents in its genome mean. That is an extraordinary place to be. Here is something I have been thinking about throughout everything we have covered. Something that is not quite science and not quite philosophy, but sits right at the edge of both. We keep using the word error to describe the mutations that built human intelligence. And in one sense, that is accurate. These were mistakes in the DNA copying process. Random changes in a molecular sequence that were not directed toward any outcome. In that sense, they were errors. But when an error produces something that has never existed before, when an accident generates a property that no accident before it generated, when a mistake accidentally constructs something that can look back at the mistake and ask questions about it, is it still right to call it an error? Or has the concept of error become too small for what it is describing? In music, an error is a wrong note. A note that does not fit the harmonic structure the piece is built on. But sometimes a musician plays a wrong note, and the rest of the ensemble hears it and incorporates it, builds on it, makes it part of a new harmonic structure that nobody had intended. The note was wrong in the context it originally appeared in, and then the context changed, and the wrong note became the foundation of something better than what was planned. The mutations that built the human brain were wrong notes. They disrupted sequences that had been functioning well for hundreds of millions of years. They interfered with proteins that had been doing important jobs. They deleted muscle fibers and rearranged chromosomes and partially blocked synaptic development. In the original context, without the compounding of all the other changes, many of them would have been harmful. And then the context changed. The environment shifted. The social structures that could absorb a weaker jaw emerged, and suddenly the jaw mutation was survivable. The dietary change toward cooked, starchy foods emerged, and suddenly the metabolic cost of a bigger brain became manageable. The cooperative child-rearing structures emerged, and suddenly the extended helpless childhood became something a community could support rather than a catastrophe for a family. The wrong notes became music, not because of any plan, because the harmonic context in which they were embedded kept changing, and the combination of what they introduced into the system with what the system already was kept generating new possibilities, kept generating new contexts in which the note could be built upon rather than eliminated. You are the music that the wrong notes built, not the error, not the accident, the music. And the music is still in the middle of being composed. Tonight, if you let yourself drift towards sleep, carry something simple with you. You are approximately 3.5 million years of accumulated accidents, each one surviving by margins that, had anything been slightly different, would have been eliminated instead. The jaw muscle. Mutation that did not kill your ancestor, because your ancestor happened to already have stone tools. The synaptic development slowdown that did not harm your ancestor, because your ancestor already had a community that could support a long childhood. The brain expansion genes that did not cause death in childbirth because your ancestors had each other, had midwifery, had the cooperative birth practices that a social species develops when the most dangerous thing its members do requires help. Each era survived only because of all the other eras that came before it, and each era's survival made the next one survivable. It is a chain of interdependencies going back millions of years, a chain of accidental compoundings, and the end of the chain is this moment. You, lying still, breathing slowly, brain running at 1/5 of your body's metabolic budget, doing the extraordinary thing brains built by these eras do, which is to keep processing even in the dark, even in the quiet, even in the spaces between thoughts. The genome is not done. It never is. Somewhere, in cells dividing right now, new errors are being made, new mutations are appearing in new individuals. The vast majority of them will come to nothing. They will drift away or be eliminated or simply persist as neutral noise in the genomic background. But some of them, the right ones, at the right time, in the right genomic context, will be something. Will add a note to a melody that has been building for 3.5 million years, and that has not yet found its final form. You are not the endpoint of evolution. No living species ever is. You are where the process currently stands, the current state of an experiment that has no predetermined outcome and no reason to stop. The errors that made you possible are still being made in you, in your children, in everyone alive right now. The process that built the human brain from mistakes is still running, silently, invisibly, in every dividing cell on the planet, not building toward anything specific, not aiming at anything, just continuing, just doing what it has always done, making copies, making mistakes, seeing what survives. And in this moment, what is surviving is something that can ask what it is, something that can hold the story of its own origin and feel something about the telling of it, something that wonders, something that rests after wondering, something that will wake up tomorrow and keep asking. The broken gene in your cells that cannot make vitamin C has been broken for 40 million years, and not once in 40 million years did any of your ancestors die from it in a way that stopped the line from reaching you. Every single one of them found the fruit or the green leaves or the roots that kept the connective tissue intact, kept the blood vessels from collapsing, kept the line going. In every generation, in every environment, through every hardship, the line persisted. The broken gene persisted with it. And here you are, with a broken gene in every cell, perfectly capable of surviving in a world full of vitamin C, heir to 40 million years of success that did not need that gene to be fixed. That is what survives. Not perfection, not optimization, just what works, in the conditions that exist, in the moment that it exists in, passed forward to the next moment. And sometimes what works is also extraordinary. Sometimes the accumulated accidents build something that looks up at the universe and begins to understand its own place in it. Tonight, you are that something, looking in, settling into the quiet, letting the errors rest. They will be here tomorrow. They have been here for a very long time, and they are not finished yet. Sleep well.